Handbook, right? Not James Coons. Okay, cool.
All right, guys, welcome to Modern Day Debate. We're going to have a fun one for you. This is going to be evolution versus creation, and the topic specifically is whether or not humans or are apes and are related. Humans or apes are related, excuse me. So we're going to do two 16-minute openings. You guys could go, 8 minutes each, or you can consolidate your time if you think one person has a better opening, and then we'll get into some 10-minute rebuttals, and then a 50-minute discussion followed by some Q&A, or 5-minute closings and then some Q&A. So I believe in email we talked about I think Austin going first.
I'm actually going to be going first this time,
So
and then Austin.
The Flying Spaghetti Auditor, colloquially known as Jed. We will start with him, and yeah, let's get right into it, guys.
Cool. Well, right on. First thing I want to do is just thank Modern Day Debate for hosting. Thanks, Raw Matt and Donny from Standing for Truth for agreeing to a discussion. So, ever notice how 100% of evolution deniers are theists? More specifically, Christian young earth creationists. Our opponents are no exception. In this debate, Austin and I will demonstrate common ancestry between humans and chimpanzees, but it's worth examining how our opponents' religion influences their denial of established science. Their rejection of evolution is not rooted in any actual facts or observation, but instead, it's rooted entirely in their religious holy book. This same book, which claims the earth is flat, bats are birds, and the first humans had a lifespan of more than 900 years. Creationists arbitrarily accept some parts of evolution, like cat common ancestry, because it's necessary for their global flood myth. They believe every single land-dwelling species on the planet fell victim to a near extinction event about 4,400 years ago. A global flood reduced the population of every animal kind down to 2 to 14 individuals. Notice that word, kind. A kind is an arbitrary taxonomical category creationists use in an attempt to reconcile the lack of space on the ark with the enormous genetic diversity of the modern world. Scientists estimate there exist 6.5 million distinct land species, which means Noah's Ark must have had at least 15 million individual specimens, accounting for mating pairs and 7 pairs of every clean animal and bird. There's no way in hell the Ark could have fit this many critters on board, so creationists appeal to lightning-fast post-flood evolution. Noah's flood would not be possible without evolution, so creationists are forced to accept it. They accept that changes in genotype are passed down to younger generations, resulting in diversification and new species. Rather than bringing all 41 species of Felidae, Noah just brought one pair of proto-kitties, perhaps Pseudoloris, which diversified right after the flood in just a couple of generations into 41 reproductively isolated species like lions, tigers, pumas, jaguars, and ocelots in less than 5,000 years. Creationists have no problem accepting this outlandish claim, along with similar lightning-fast diversifications for every other animal kind, except humans. Even though there is more genetic dissimilarity between cat species than between ourselves and the other great apes. Cats took about 16 million years to diversify, while our split with chimpanzees was only about 7 million years ago. So why do they special plead when it comes to human evolution? Well, it's obvious. They are humans, and they have a personal bias to reject the overwhelming preponderance of evidence because it hurts their ego and contradicts their nonsensical book of fairy tales. And I'm going to go ahead and pass it over to Austin. Thank you.
Trying to work on
it. All right, just let me know when I can share my screen.
Hold on, he wants to share his screen.
Alrighty. Now, yep, I just hit it. Is it sharing? Cool. Alrighty. Let me know when my time is starting. All right, so the topic of the debate tonight is, are humans and apes related? Which you might notice is kind of like asking, are chihuahuas and dogs related? Are ducks and birds related? Are tigers and cats related? Humans are apes, so to ask if humans and apes are related is a little non sequitur, but we'll go over the data a little bit, and that's what I put this presentation together to do. So the first two slides are just about the history of evolution here. The main reason I've included them, there's a lot of good information in here, so if you're watching this back, pause it and read this. But the main reason I included them is to illustrate the point that between this era and the first slide here, we're already debating immutability and mutability. There's people like Anaximander saying species might be mutable. He claimed that was because he didn't see as a real possibility that human babies could survive in the wild. But people were putting forward theories of mutability for a long time. And this was because we could see mutations happening in the wild, but we could see organisms being born that had albinism or melanism. We could see all kinds of mutations, but we didn't think it was possible for random mutations to build new features and entire new body plans. And that changes by the time you get to this second slide, era 3. That's basically what Darwin and Wallace figured out was natural selection, the non-random aspect of evolution. That natural selection was non-random. Just by being difficult to survive in, the environment acts as kind of like a selector, like what occurs in artificial selection. By not having enough for everything to survive, it selects the most fit organisms. And then we knew that, a random mutation can build new features and novel body plans because of non-random natural selection. So as far as the evidence that humans and apes are related, there's a blinding amount of it. It's overwhelming. As far as the evidence, I'm going to put forward 5 main points tonight. Those are morphology, genetic similarity, the second chromosomal fusion, cytochrome C, and endogenous retroviruses. So let's start off at morphology. So as you can see in this diagram down here, we've got a bunch of humans. There's some anatomical modern humans and some arcane humans. Donny and Matt are basically going to have to argue that there's no real significant differences between these arcane humans and the modern anatomical humans, that they're all either modern humans or chimpanzees. I'm not sure exactly what their argument about this data is going to be, but I would remind you over and over again that this is almost absurdly data that better fits a model of common ancestry and speciation, species mutability, than it could ever fit a model where species immutability is in any way present. And then, of course, we have all of the human chimp body plan similarities: the heads, arms, legs, chest, pelvis, bones, tendons, muscles, joints, brain, heart, lung, stomach, intestines, all present, all in the same place, all doing basically the same thing. Their behavior, embryology, and development, all very similar. Tool use, emotional expression, social structure, and self-recognition. These are things that make a large portion of this chart over here humans. And then these last 3 points are the most substantial points for morphology. The mammal primate dentition, another part of what makes a lot of these humans the Y5 molar plan. The migratory foramen magnum, that's opening an aperture on the skull that migrates from a posterior position to an anterior position as you move up this chart. And then another especially strong point are vestigials, the tailbone, which can be safely removed surgically if needed. And some humans are even born with atavistic tails. You can actually go to Google and look up a picture of that if you want. I decided not to include one because it's pretty much all naked babies, newborns. It occurs due to something called apoptosis. The way that fetal development plays out is that cells in the fetus are instructed to grow. Apoptosis tells them to stop growing. Sometimes the genome inadvertently expresses an ancestral form. The tailbone, the tail portion of the fetus isn't instructed to stop growing. In fact, all humans grow a tail in fetus form. It's just most of us absorb it. Some people don't. Go look up a picture of that if you've never seen it before. It's fascinating. And then there's the genetics. This is the what has kind of been like the Galileo moment with evolution. We have so much genetic data now, and because it's in the form of nucleotide identities, you can do statistical analysis on it. And so when we compare genomes, we can see that the genomes are readily available. You can go look at these genomes and sift and shovel through them yourself. Humans have 23 pairs of chromosomes. Other primate species have 24, so very close to the same number of chromosomes. The number of genetic base pairs is also very similar across several species, 3.1 to 3.3 billion genome base pairs on humans, chimps, and gorillas. This makes them all very highly comparable. And then the actual numbers, you got protein coding base pairs at 98 to 99% similarity between humans and chimps. Telomere to telomere, it's 85 to 86%. We see centromeres inside of the human's second chromosome, indicating that those were once the ape ancestors' second and third chromosome. We've got cytochrome C, which has very few amino acid differences between even the most different forms of life, and zero amino acid differences between humans and other primate species. And then the ERVs, those are the best evidence, so we'll save those for last. You et al., I know this paper is going to come up, so real quick, I'm going to touch on it. The most important thing to touch on is that it did not change the number of protein coding base pairs. It's not like it went from 99% similar to 86% similar. We're still 99% similar in protein coding base pairs. 86 is for a different method. It also showed that intraspecies difference numbers weren't significantly higher from intraspecies. That means you can be more different from another human than you are from a chimp. That shows common ancestry very conclusively, and Casey Luskin told the most transparent lie possibly ever told about this and was caught red-handed in and had to retract it with egg on his face. So then the second chromosomal fusion. Humans have 23 pairs of chromosomes. Other primates have 24. We made a bold prediction. There'll be a centromere inside. There'll be signs of a genetic fusion in the past. We found those signs. Great example of what's called scientific evidence, bold prediction bearing out as true. Cytochrome C is another example of genetic evidence that humans and primates share ancestry. Cytochrome C is a gene that produces a sequence of amino acids that's highly conserved. It's super important for processing nutrients and developing energy for your body. So that's why it's highly conserved. And it's also the host for evolution because improvement in being able to process nutrients is obviously something that's heavily selected for. But even still, there's not that many differences. And the best part about it is the differences are very small. It's very easy to understand. And those differences perfectly recapitulate taxonomic phylogenies. Chimps and bonobos and humans have zero differences. Gorillas, orangutans, rhesus, and humans have one difference. A gibbon has two. You need to get outside of the entire primate category before you get any more than three differences, and all the differences outside of that also match taxonomic phylogenetic classifications. Then there's the endogenous retroviruses. How much time do I have left, Mike? Okay, so I'll try and rush through this as much as I can, but it's truly a new Galileo moment in evolution. It's making it very difficult for someone to reasonably and rationally deny common ancestry and macroevolution. I think it's virtually proof because, again, this statistical analysis, you can have well over 99.9% confidence that the endogenous retroviral insertions that exist concordantly in humans and chimpanzees and bonobos. The odds of that occurring by random insertions is like well over it's a number smaller than 1 over a number equal to the total number of atoms in the observable universe, a number that's incredibly small that asymptotically approaches 0. So you can be over 99.99999% confident that these ERV insertions come from common ancestry and macroevolution. And what an ERV insertion is, ERVs are viruses. They can attach to a host cell. They use a special enzyme to break the host cell open and inject their own chromatin into it. And when they do, they leave behind this, they do it in a germ cell, like a sperm cell or an egg cell. That organism, once developed, it will have that DNA in its genome, and these have been accumulating over evolutionary time. We know they're viruses because we've re-engineered some of them. We've taken the virus sequence from in our DNA, fixed the mutations, and reverse engineered a virus. We called it the Phoenix virus. It acted like a normal virus. It was infectial, took over a host cell and made it produce the viral product. We can also know that they're ERVs because of mammalian versus viral codon bias. This is the way DNA produces proteins. DNA produces proteins differently in viruses than it does in mammalian. In the rest of the mammalian genome, the segments that are outside of the long terminal repeats that contain the endogenous retroviruses, we see mammalian codon bias. Inside of the endogenous retroviruses, we see viral codon bias. We're watching an endogenization event happen right now. It has almost completed the arc of endogenization. It's called the koala endogenization event. The koala retrovirus is currently becoming fixed in koala populations. And the source for all of this information comes from a evangelical Christian preacher guy named Graham Finley. So it's not like it's mean secular science. It's a Christian man, very devout. And I've got some questions for Donny we can get into in the open discussion. I know he interprets ERVs as designed elements, so I got some questions for that. And this is just a fun little slide here. This is a little mirth because my presentation was kind of bland. This is Matt, me, and Donny as Australopithecines having a little chuckle at the Bible. And that is the end of my presentation. I think my time is pretty much up there, right? Thank you.
Okay. All right. Let me get out of here. Perfect. Oh, no. Did everybody get rearranged? How did that happen? All right. Are you sharing a screen, buddy? Okay. That's why it was messed up. Give me 1 second.
Yeah, give me 1 second. I just want to grab my timer as well, make sure that I'm okay. I'll start the timer right now. Okay, thank you, gentlemen. I enjoy that opening statement. Many of my favorite talking points and arguments to refute. So here we go, debate. Are humans and chimps related through common ancestry? I love this topic because in many ways we can refute or overturn the entirety of common descent by answering the question, are humans and chimpanzees related? Because if we can demonstrate that humans and chimpanzees are not related and that they can't be traced back to a common ancestor roughly 6 to 7 million years ago, then that means humans and strawberries or humans and banana plants are definitely not related. So I like to stick as close to home as possible. The evolutionist argues that chimpanzees are our closest cousin and that we can be traced back to a common ancestor roughly 6 to 7 million years ago. And so let's show why that is not true. Let's start with the Y chromosome. Now, our interlocutors here, as you can see already in the opening statement, they want to focus on all the similarities, but then ignore or dodge the major differences. I want people to remember this. It's the differences that make all the difference. We can explain similarities as common design proponents. Evolutionists can explain the similarities, but it's the differences that make all the difference. When we have massive discontinuities or dissimilarities, that's where the evolutionist struggles. But that's where we excel, because that's exactly what we would expect if separate ancestry is true. So notice this in the conventional literature. So we got the Y chromosomes of humans and chimpanzee, or of the great apes. And when you consider overall architecture, size differences, and gene content, the human Y and chimpanzee Y are less than 30% the same. You can see that the chimpanzee Y is half the size. It's lost apparently the entirety of its heterochromatic arm. How do our interlocutors explain that? Heterochromatin is dense, tightly packed DNA that used to be considered inert, but we know that it plays vital roles in nuclear organization, chromosomal stability, and gene silencing. Now, this again, right from the conventional literature, despite a recent divergence of these species, and so this was not expected or predicted, their Y chromosomes differ just a little bit, no, enormously in size and gene content. As we went over, the chimpanzee Y is only half the size as the human Y. And in terms of the so-called nested hierarchical pattern in the great ape family, the human Y is more similar to the gorilla Y than to the chimpanzee Y. They admit this directly in the literature. Now, it gets even worse for evolutionists. 2024, we have the most sophisticated sequencing data for the Y chromosomes. And it turns out that the differences in the Y chromosome between humans and chimps is even more. Again, it's roughly 26.2%, less than 30% the same. Now, based on this data, we have some questions for our interlocutors, because there's massive differences in heterochromatin. We have differences in palindrome sequences, ampliconic sequences. And so here are some points that our debaters are going to have to address. There's no ancestral Y chromosome reconstruction. Evolutionists cannot claim gain or loss since the split without knowing the ancestral state. The 2010 paper itself said the ancestral state cannot be determined. And so my question is, can they reconstruct, or have they reconstructed the Y chromosome ancestral state in order for us to determine what was lost, what was gained, in order to account for these massive differences if humans and chimps really are related? There's no rate-based population genetic model. Not one critic, and maybe our debaters here, our interlocutors, can produce this here tonight. There's no model showing the required duplication/deletion rates, required recombination frequencies, timeline feasibility, so on and so forth. The structural differences are too large. Refute me on that. Evolutionists, like in our last debate with Planet Peterson, all they do is provide mechanisms, just so stories, evolutionary philosophy, without actually demonstrating that those asserted mechanisms, like, well, duplications, deletions, faster rates of substitution, sperm competition. Okay, that's your evolutionary philosophy. Now you need to model them, quantify them, test them, simulate them. And so here's some questions for Austin and Spaghetti here. How do these massive architectural changes occur mechanistically in 6 to 7 million years? When did these changes occur within the proposed 6 million year time frame? Why did they occur? For example, what selective pressures drove or tolerated them? How is the entirety of the heterochromatic arm lost in the chimpanzee Y? We have the 2024 study, Makova study, which gives us high quality Y chromosome assemblies, but it does not do a few things: reconstruct the ancestral ape Y chromosome, identify directionality of the changes, gain or loss. And so this is a major problem for our interlocutors here. Here's another question I really want answered here tonight. They need to meet their burden of proof. Are humans and chimps related? Okay. Which lineage lost what? What specific amplicons, palindromes, or gene families were supposedly lost in each lineage? It's also very interesting that when you compare Y chromosomes, Y chromosome is uniparentally inherited. It's inherited on the father's side, so paternally inherited. You could take all Y chromosomes worldwide in human males. There's extremely low diversity. We're about 99% the same in the Y chromosome. The mutation rate is fast. It's roughly 1 to 3 per person per generation. And there's low diversity, which means you can trace back all Y chromosomes to a single Y chromosomal ancestor roughly 4,500 years ago, who's nothing like the chimpanzee, nothing like the great apes. And so again, show me the details, model the types of losses that took place. Where is the ancestral Y chromosome reconstruction? If loss is invoked, you must show the ancestral state. But the 2010 Nature paper explicitly states the ancestral Y cannot be determined. Without an ancestral reference, loss is just speculation. Why do humans have entire palindrome families and gene systems absent in Pan? How does a 2-time size difference arise purely by loss? So we could start with those, and we'll see if they can answer any of those challenges. Now, evolutionists will oftentimes ask the question, well, if humans and chimps are related, if evolution were true, what would you as creationists expect? Well, I would expect that the genomes of humans would reflect a long history of evolutionary change. So we should expect the genomes of humans to be largely junk, reflecting evolutionary leftovers, genetic mistakes, viral fossils. But if creation's true, if design is true, then we should expect a genome that's near fully functional. Well, the ENCODE project and subsequent projects have actually demonstrated that over 80% of the genome is active, suggesting function. We've got many papers showing functional roles for so-called pseudogene RNAs. What they do is they produce RNAs that regulate protein coding genes. They act as decoys for microRNAs, protecting functional RNAs from degradation. Many papers there. Consider the functionality of the genome. What was once called junk DNA is now known to be extensively functional, regulating, coding, and organizing. This is a bigger problem for the evolutionists tonight because the drivers of evolutionary change, selection, mutations, gene flow, well, that means the majority of your neutral mutations are actually nearly neutral, slightly deleterious, because the more functional the genome is, the more deleterious these mutations are going to be. Austin talks about endogenous retroviruses. We can get into that. The question is, are these really the ancient vestiges of past viral infections, or are they created units of DNA function? This is one of my favorite papers in 2021. He says, well, we know they're viruses because of their genetic makeup. They share sequence similarity to exogenous retroviruses. They got the GAG, POL, ENV components or genes. You got the LTRs, long terminal repeats. Well, it turns out that they require that specific genetic makeup to carry out many of their functional roles. One of them, protecting us against viral infections, acting in the immune system. They also perform viral mimicry. Notice this. Why do we share ERVs with chimpanzees and the great apes? Well, HERVs appear to play important roles in physiology and development. Notice this. Yes. Yeah. 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 He's going to get about 5 minutes. I'm taking about 11. So I'll start wrapping up here. If the accidental infection, notice how they assume an infection took place, of a mammalian ancestor by an exogenous retrovirus had never occurred, the placenta and the mammals, notice this, that produce it, including humans, would have never existed. So we actually would not exist if it weren't for the existence of endogenous retroviruses. And so why do we share endogenous retroviruses with the chimpanzee and the great apes? Well, we both have embryological development. It's like asking the question, why do all modes of transportation share an engine in common? Well, if vehicles didn't have an engine, they wouldn't be getting very far. And so paper after paper here answering the question of ERVs. And he also talked about the chromosome 2 fusion, cytochrome C, but we'll get into that in our rebuttal. Matt, you got roughly 5 minutes, brother. Go ahead.
All right. Just got to share my screen now. The host isn't allowing multiple. Oh, can you take yours down? I can't share until yours is gone. Yeah. Okay. All right. Everything good?
Yes.
All righty. I also agree that any model should put their predictions to the test. For this one, we're going to be talking about mutation rates because over time, mutations increase in a population, and that's how we count and determine, the diversity on life on Earth. It should be increasing and continues to increase. So I believe mutations are the most important aspect of all of this because that is how we answer that question. Evolution says and predicts a slow mutation rate based on the fossil record. Creation predicts it to be fast because of the biblical timeline. Evolution takes a split into consideration, and they use this to calibrate called the phylogenetic mutation rate. We use what is known as the pedigree observable germline mutation rate. What did we actually find when we tested this and we ran the studies? We found the rate was 200 times higher than the evolutionary phylogenetic rate. We found a rate of 1 every 25 generations, a substitution arises. Evolution expected 1 every 590 generations. This was the first pedigree study that was done. It was 11 generations deep by Howell. He confirmed Ludstorm's mathematical equation in 1992 that suggests that a much more recent common ancestor of humans, about 1 mutation every 25 generations, places the most recent common ancestor very, very recent. Then we go to 2024. Agnar Helgeson did a study, and he actually looked at the entire mitochondria, the coding region, the control region, and the HVR, and found something amazing. All of those different regions have a different rate of change. But what's going on? How is that possible? Well, he found that the coding region was only accurate within about 20%. The rest of the time the clock was ticking, it was inaccurate because selection is occurring there. However, in the D loop, what he discovered is that it was 8 times faster than comparing it to the coding region. So that means the clock is 90% accurate with some wiggle room of about 10% where there might be some selection happening. Then he looked in the hypervariable region and found it was even more accurate. And there was only like 40 base pairs in both hypervariable regions even mutate. And mostly they are conserved. They're neutral. So Parsons came along, and he works for the FBI in his blood bank, and they tested it. And what did they find? Well, they compared the DNA sequences of the two control region hypervariable segments. And guess what? Most studies do this. All of these studies look at the D loop or the hypervariable region. They're not looking at the entire mitochondria where selection can be occurring. So when they look in the mtDNA D loop, they said about 5% mutations occur in that run region and 7% in HVR2, but they're functionally neutral. So therefore, the clocks that they tell us are very accurate. Entire mtDNA, not so much the case. But in neutral regions, the rate at which a new mutation arises is the same rate which becomes fixed. So therefore, the substitution rate becomes the fixed rate. So he determined and published 6,500 years ago was around the time that mitochondrial Eve lived. And a lot of people say, well, his rate was really fast. It was only dyad studies. Actually, no. He studied some of those. They were from the AFDIL bank. His pedigree depth was only about 1.7 generations, but he didn't publish that rate. He published the rate that was in the deepest pedigree, which was about 6.4. So the reason we like Parsons is because he studied the most diverse groups of people. And that's kind of what you want to do when you want to get a global population-wide substitution rate, right? So he had some critics, and they came and joined him the following year to determine could this even be right at all. And Holland joined him in 1998 and confirmed it again. Actually got a faster rate, 1 every 30 generations. His rate is used to this day in real-world applications by the FBI. Not the evolutionary rates. They actually said that the observed substitution rate from both blood and cell samples greatly exceed evolutionary inference times. And guess what? We only find 14.1 fixed substitutions total in the entire region. That's not very many for how fast the rate is. So again, they said this overall mutation rate with both of our studies came to a remarkably similar conclusion, 0.25 and 0.28 between Parsons and Howell. But then much later, we have the deepest rooted pedigree study yet. It goes back to the 1410 for the most recent common ancestor. Magradol got the exact same rate, yet again, from the deepest pedigree study that they've ever done. So time didn't affect it. And this was confirmed again in 2022. She can 16 times faster than the phylogenetic estimates, about 1 every 38 generations. And then going directly back to that original study, 2024, Helgeson, he also got exactly the same rapid mutation rate that lands on an Eve right around 6,500 years ago. The time changes based on generation time. Parsons used 20, he used 30. Hayer got the same results. She tested HVR1 and getting 2, and again, same thing. She used a 29-year generation time, and she got those 2 rates up here. They basically are the exact same time as Parsons. So the question is, why are these rates wrong, and why do the experts keep getting the same results? Selection isn't interfering because they're neutral mutations, and even they admit this. They said selection is unlikely to be a major factor that underlie the differences between these. At some points, the results suggest that selection has not preference.
Matt, I think you got about 30 seconds.
Okay. All right, no problem. Sure. It's rather the phylogenetic rate that must be the biased, and it is clear that the pedigree rates are the mutation rate. So if the mutations are the basis of evolution theory and it can't get those right, what does that tell you about the rest of the theory? They admit that there should be more diversity, but there's not. It's not there. Good enough.
Yeah, that'd be perfect.
Get back to
How's
the
my
original
audio? Do I still
scene.
have a weird echo?
Let me double check real quick. The chat will let you know here in a second.
Nothing worse than a weird echo.
All the screens got scrambled. Great. Okay, let me just try to get these back in the order that you came in on. Standing for Truth is in the top left-hand corner. Oh, this is terrible. This Zoom shit just completely messed everybody up. Might honestly be worth it to get to this scene here.
Perfect. So demonstrating that chimpanzees are not our closest relative, if you were able to do that, would not necessarily debunk all of evolution or prove that we're not related to strawberries. Scientists can be wrong about evolutionary lineages. We used to think that elephants and hippos were very closely related, but now we understand that hippos and whales actually share a more recent common ancestor. Similarities equal common design. That's just an appeal to magic. Donny says differences debunk evolution, but differences are exactly what we would expect to find under the evolutionary model. That's exactly how evolution works. Mutations create differences in populations which accumulate over time. The process of sperm production includes a huge number of cell divisions. This is with regards to the chromosome Y. Every division is an opportunity for mutations. It lacks a recombination with a sister chromosome, which prevents many DNA repair mechanisms from efficiently fixing errors and leads to an accumulation of mutations over time. Discovering junk DNA that actually serves a purpose is not a reputation of evolution. The human body often repurposes junk DNA to serve a purpose. There's actually an endogenous retrovirus, which served as the foundation and development for the function of placentas. So if it weren't for ERVs being repurposed, placentas wouldn't even exist. Donny claims ERVs were placed in the same location in humans and chimp genomes by divine design. Now, notice when it comes to the most clear-cut and obvious evidence for evolution, his only option is to appeal to magic. Anything is possible if you appeal to magic. Creationists make an arbitrary distinction between micro and macro evolution. They say evolution is possible within a kind, but not from one kind to another. They assume there's some kind of mechanism which prevents organisms from evolving too much, but they're incapable of demonstrating how it works or clearly defining where the boundaries are. There's actually a bigger difference between felid species, or cats, than hominid species. Ocelots, for example, have 36 chromosomes, while most other cats have 38. Pumas and jaguars both have 38 total chromosomes, but jaguars have 34 bibrachial chromosomes and 4 teleocentric chromosomes, while pumas have 36 and 2, respectively. As a result, ocelots, pumas, and jaguars are all incapable of producing viable offspring with each other. This means speciation and chromosome mutations are not boundaries for microevolution. Donny and Matt accept that cats share a common ancestor, but reject hominid evolution despite cats having more dissimilarities. Every argument they apply to human evolution can also be applied to cat evolution to an even higher degree. This demonstrates our opponents' inconsistent levels of skepticism when analyzing the ancestry of humans compared to other organisms, showcasing their human bias. And I'll kick it over to Austin.
All right.
Thanks, Jed.
Austin, go ahead. You actually have 2 extra minutes if you want to use up the rest of his time. So
Thanks.
7 total.
So, I'm having a great time. I hope the audience is enjoying this too. This is a fun debate so far. Matt and Donny made a lot of points in their presentations. I took some notes. So the Y chromosome. The Y chromosome is the way it is for characterizable reasons. We know that the Y chromosome has a lot less genetic base pairs in it than other chromosomes, and that a large portion of these genetic base pairs are dedicated to the process of spermatogenesis. And this helps explain why the structural and gene base pair differences are the way they are. It's because we come from promiscuous ancestors. Our female ancestors would have sex with several males, and so the sperm would have to compete for the right to pass on their genetic material. And this is partly from competing with other males, and it's partly to do with the way the female reproductive anatomy naturally is. The vagina is very acidic, it has a pH, it has physical obstructions that stymie impregnation. And this in itself contributes to the process of selection, only selecting the fittest sperm with the most motility, the strongest protein coating. When you see that you have organisms with a wide variety, you're going to have the greater outliers. You're going to have some sperm that are much, much more significantly motile, much better coated. The outliers of variation are what produce that difference in the Y chromosome. And I think it's kind of ironic because we're pointing at these differences in the Y chromosome and saying, oh, you can't explain this with common ancestry. But what we're really pointing at is derivation. We're looking at the Y chromosome being derived by evolution. It's exactly what we're looking at there. Let's see here. Disconfirm. Oh, right. Okay. So this is less rebuttal towards a specific point and just a general criticism, which is that the way the scientific process works, not knowing something isn't disconfirming data. Disconfirming data is what would cause problems for evolution. Not having an answer to every single question doesn't really cause a problem for evolution. If there's data that doesn't fit evolution, that's something that causes a problem. Like when we looked in the geological column, we had found that at the deepest layer of fossil strata, all modern extant and extinct organisms all appear horizontally at the same time, anatomically modern humans and arcane humans as well. That would have been data that disconfirmed evolution. It would have even been data that supported a creation model. But obviously, that's not what we saw. And especially, we saw arcane humans. There's millions of arcane humans before a single modern, anatomically modern human fossil can ever be found, which is devastating to our opponent's arguments. Junk DNA, Jed already touched on that. The mutation rates. So Matt's talking about the scientific process working. He's talking about making a prediction. We expected something. We found data that didn't quite fit the prediction, but could still be explained. And what it doesn't especially do is it really doesn't fix the problems in the evolutionary rate. Even if organisms are evolving 16 times faster than we thought, it's just arranging deck chairs on the Titanic. It doesn't even come close to making up the difference between the actual timeline and what the young Earth creationist is proposing. And let's see here. Basically, so not only does it mathematically not solve the problem, observationally, it doesn't work. We don't see this rate of hyper change happening. So to make this model fit, basically you would need a new species of elephant every single generation. And as far as I can tell, elephants give birth to many, many, many elephants of the same species before any sort of speciation happens. So it's not only, it doesn't mathematically fill the gulf, observationally, it doesn't make sense. We would see rapid evolution. We don't see that except with things like artificial selection in microorganisms and insects, species that have extremely fast generation times. Their lifespan is short. They produce a ton of offspring. Their, developmental cycle is very short. Am I running out of time? How much time
You
do I got?
have 50 seconds left.
I think I can stop there. I think I covered all the points that I wanted to get to. So I
Okay.
don't want to ramble.
Sounds good. We're going to kick it over to creationist side here in just a second. Oh, are you trying to share your screen again? Gotta let
Yeah.
me know in advance because that's messing
Oh.
up the Zoom call.
Apologies.
All right.
Let me stop share then and then redo it.
No, just give me a second.
Okay. Okay, you're doing a great job. I appreciate it. Okay, here we go. We'll start the timer now. Okay, well, those rebuttals did not answer a single question that I asked on the Y chromosome. All they did was confirm my prediction that what they will do is list mechanisms. They'll list mechanisms to explain the Y chromosome dissimilarity, but no model. Where's the Y chromosome ancestral sequence? So we can actually model what they're saying. They're just giving their evolutionary philosophy. All they've done is demonstrated to the world that they're really good at what? The 5 Ds of dodgeball. Evolutionist dodgeball, dodge, duck, dip, dive, and dodge. So the first question I want answered in the discussion is how, why, when, and where was all of that heterochromatin lost? The palindrome sequences, the ampliconic sequences, and show me the ancestral Y chromosome sequence. Show me the reconstructed sequence. Okay. Endogenous retroviruses, another dodge. Okay. If they're going to say that their interpretation of ERVs is superior to mine, then they're going to have to answer, where do all these functional roles come from? Embryological development, where if we did not have ERVs, we literally couldn't exist. Here's one question I want Austin and Spaghetti to answer. What is the evolutionary pathway by which an LTR, long terminal repeat evolved? LTRs contain enhancer modules, promoter architecture, TF, that's transcription factor binding clusters, chromatin opening signals. Show the mutation by mutation pathway. We're not interested in your evolutionary philosophy here tonight, okay? Here's some more questions. How did the preloaded regulatory functions inside ERVs arise in the first place? Okay, the functions associated with the integrase, the reverse transcriptase, the LTRs, their functional roles in determining cell types, acting in the immune system, acting in embryological development, for example. How does random viral insertion produce coordinated developmental timing? Answer that. It was just a complete dodge. And it's so funny that Flying Spaghetti Monster says, well, if you disprove the idea that humans and chimpanzees are related, well, that doesn't mean common descent is falsified. Dude, if humans and chimps aren't related, then by definition, special creation's true. So who cares if humans and strawberries are related or not, okay? All we have to do is show you that humans and chimps aren't related. That answers the phylogeny challenge. He starts the debate by bringing the Bible into it right off the bat, complaining, calling it a bunch of fairy tales. I mean, that's a weak start to an opening statement. He's not meeting the burden of proof. The Bible, he talks about the Bible teaches a flat earth. Well, that's his assertion. Okay, the Bible talks about God sitting upon the circle of the earth, and Job says God hangeth the earth upon nothing, a clear reference to gravity. He scoffs at man living to 900, even though we got plenty of scientific points of evidence showing that man could and did live to 900. Not the topic. It's just interesting that he brings those up. Kinds. Yes, the best definition of kind is groups of organisms that can be traced back to a common ancestor. We can do this with humans, which refutes their position tonight that humans and chimpanzees aren't related. One piece of evidence is what? Well, the fact that the Y chromosome dissimilarity cannot be answered. He misrepresented what I said. Yeah, evolutionists can explain some differences. They can explain similarities and differences. Okay, since we as humans are so similar to the chimpanzees on the outside, I would expect that on the inside, we're quite similar as well. But we need to remember, it's the differences that make all the difference. Focusing on all these morphological similarities like Austin did, but ignoring the massive dissimilarities, okay, that's going to be a problem. That's them not meeting their burden of proof. Austin talked about the chromosome 2 fusion. Well, he also talked about cytochrome C hierarchies, ERVs, which we already addressed. Okay, chromosome 2 fusion is interesting. I've got a couple articles that I've written on the topic, and I did a research project looking for similar signatures that we see in the chromosome 2 spot. For example, we find TTA, GGG, CCC, TAA in a head-to-head organizational pattern in the so-called chromosome 2 fusion site. Evolutionists have argued that that's the only spot in the genome that you find those signatures. We actually discovered them in several other chromosomes, the chromosome 9 being one of them, a very similar signature head-to-head organizational pattern. And yet we know the chromosome 9 was not the result of a fusion. But these interstitial telomeric repeats are functional in gene regulation, for example. But even if there were a fusion, that person in the evolutionary model who would have experienced that unilateral mistake, this would be a near extinction event. They would be the ancestor of all people on the planet. And so my question is, how could you fix that unilateral mistake in all people? That means that one person now has 46 chromosomes. Every tribe, every person on the globe eventually has to be wiped out, all their lineages wiped out, in order for there only to be the 46 chromosomal signature. Okay. The better model in explaining a so-called fusion, if there was one, would be the biblical model, where we actually have a bottleneck at the flood and at the Tower of Babel to explain how that could become fixed. The cytochrome C I'd like to address, and then we'll hand it over to Matt. So cytochrome C, highly conserved protein, falls out into nested hierarchical patterns, but he assumes that the cytochrome C has only one primary function and no secondary function. So yes, the primary function of the cytochrome C, that shows the similarities across the biological world, but the differences would be based on secondary functions. And creationists, going back as far as 2013, have actually looked to phenomenons like protein moonlighting, where you have these highly conserved proteins, mitochondrial DNA proteins, that have multiple functions. Okay. And so where you look at the secondary role, that's more related to information flow. Well, information flow is going to be different between humans, chimps, dog, fish, yeast, and therefore we'd expect differences and expect hierarchical patterns. We could get into all of the fossils that they talked about with the Australopithecines and Neanderthals and so on and so forth. There are major discontinuities. Okay, for example, the ischium in the pelvis of the Australopithecines versus the ischium in humans. Humans, it's short. In Australopithecines, it's long. So yeah, they walked upright, but it would have been a different or unique bipedality to us as humans. The hyoid bone, for example, ours differs from apes and the Australopithecines, where ours is designed to give us the ability for sophisticated language and speech, whereas the Australopithecine hyoid and the extant great apes, theirs is built completely different, showing another discontinuity. So there's enough differences in fossil data to suggest that the Australopithecines are unrelated to us as humans. Matt, you have about 2.5 minutes. I'll stop sharing in case you wanted to share. Go ahead. And you're on mute, Matt.
Thanks. 2.5 minutes. He got me good on this one. Okay, Carl Linnaeus. He was the one that introduced these nested hierarchical patterns of biological classification, but he was a creationist. He came up with this idea, and he's the one that placed humans next to primates without any evolutionary relationship at all. So just to say that, well, humans are apes, therefore we are related, that's not even remotely what he was doing. So next, we have Professor Sir Richard Owen. He was the father of homology. He also made predictions that there would be a hierarchical pattern as well. Darwin actually took this and said his similarity due to common design, and he changed it to similarity due to common ancestry. So it was just moved over and shifted to look like evidence for evolution when it was always a creationist prediction in the beginning. Then we find the exact same thing with Ernest Mayr here and creationist. What was his name? Dr. Henry Morse. They predicted totally opposite things. He said the search for homologous genes is quite futile, except for very close relatives. Why? Because mutations scramble information, so over deep time, nothing would actually be related. Henry Morse did the opposite. He said the same similarities are predicted on the basis of a same common purpose designer. It may yet be determined that all living organisms exhibit similar underlying DNA level properties reflecting unified patterns of functional design. This was before they even knew that there was function going on in these sequences. Who ended up being correct? Well, in 1977, the first universal RNA phylogeny was discovered in bacteria, eukaryotic organisms, and guess what? They shared the same, confirming biblical creationist predictions. Then in 1978, the following year, he discovered Hox genes in the fruit flies, shared among everything. Again, no biologist had even the foggiest notion that such similarities could exist between genes of such different animals. It was inescapable, confirming biblical creationist model, actually, and yet evolutionists now use it as the best evidence for evolution when it was our predictions. Genetic boundaries exist, and this is something we'll have to explain because I have less than 2 minutes. Okay. 2 slides. Wow. I don't know where to go. I'm sorry. I don't think I could do much with 2 slides. Okay. And this gene study discovered genetic boundaries. What that means is that looking at a sequence, do things converge going backwards in time? Like, for example, foxes, do they converge going backwards in time to wolves and dogs? Are they all related? Young Earth creationists might say yes, all dogs were, like all cats were. I've run these numbers, and they're not actually. Small cats and big cats are not related. Foxes are not related to dogs and wolves, but dogs and wolves are. So yes, we can talk about that. And I actually have a study on this, which will be on the Standing for Truth website soon, because I actually ran the barcoding for all cats. So good enough.
Back without me having to redo it. That's just like what Ryan would do right there.
You're doing good. 10 out of 10.
All right. Okay, so we got the 50-minute open discussions. I'm going to start the timer now. Like I said, get your super chats in so that we can have a stack of them for the Q&A. I think that'll be probably the funnest part for everybody if we can get some good ones. And then we'll start the 50-minute open discussion now. And whoever wants to ask the first question, I'll let you go first.
Austin, if you don't mind, I appreciate your opening statement and rebuttal. Can I share screen? And I know you have some questions for me on endogenous retroviruses, but I do feel like you guys did not. And if I could screen share, perhaps, Mike. So there's a few more significant questions here. And what I saw from you guys in terms of your attempts to answer the Y chromosome dissimilarity challenge is providing mechanisms, which again is fine. You know, we know these kinds of changes exist: duplications, gene deletion, substitution rate, sperm competition in chimpanzees, for example, can drive mutational changes. But let's start with this question, if you don't mind, Austin or Spaghetti, whoever wants to answer it. I'd be curious as to your thoughts on this. If you're going to invoke, well, okay, we have these mechanisms, but then not model them out, because that's what I'm looking for. Looking for is a sophisticated mathematical model or a population genetics model that is actually showing when these changes occurred, how they occurred, why they occurred, in how many they occurred in. And so this question here on screen, where is the ancestral Y chromosome reconstruction for us to even test if what you're saying can account for the differences? Go ahead. Well, if they want to look at the question, but if they don't, then we can stop the screen share. So it's up to you, Austin. Do you want to look at the question, engage it, or do you?
We can leave it up. That's fine.
Okay.
What exactly? Were you want to know what a previous version of the Y chromosome looked like?
Yeah. Well, so today we have humans and chimps. You're arguing that chimps are our closest cousin and can be traced back to a common ancestor roughly 6 to 7 million years ago. All right. So if I go to this slide here, it's a good clarifying question. So if we go to this slide here, very interestingly, we have the human Y and the chimpanzee Y. Chimpanzee Y is half the size. It's missing the entirety of its heterochromatic arm. As expected, if chimps and bonobos are related, the bonobo is also missing the entirety of its heterochromatic arm. Heterochromin is important. It's functional. I went over that in my opening. And so the Y chromosomes are less than 30% the same. So you're going to have to explain how these changes came about. You're going to have to model them. Here's your common ancestor, roughly 6 million years ago, right on the screen. So you're just invoking mechanisms, you and the spaghetti both. That's interesting. It's evolutionary philosophy. Okay, I mean, you could do that. But where's the ancestral Y chromosome sequence here for us to even test if those changes took place? That's my question.
Yeah, and Austin might be able to know more about this than I do, but my understanding of genes is that they don't leave fossils. So if you're looking for us to make a completely accurate reconstruction of a gene going backwards in time, I'm not aware that that's even possible. All we can do is speculate about what genes looked like in the past, but we're never going to be able to replicate like a Australopithecus Y chromosome because we just can't do it. It's not possible. We don't have access to the information to look back in time.
Is it your position that the ancestor would have been an Australopithecine before the split?
No,
I would say something Australopithecine-like.
Okay, so what's the best? And yes, you can do it. I mean, you could look at the differences between humans and chimps. You can put out your mechanism. And I appreciate you guys conceding the fact that, again, all we get are mechanisms, just so stories. But we can't even test that if you're not going to provide a sophisticated mathematical model in terms of how these changes occurred, when they occurred, why they occurred. And so, yes, you can do the work, or a scientist can do the work to get a close approximation to what that hypothetical Y chromosome ancestral sequence looked like. And then from there, we can model it out. So, okay, you say we can't get an exact one. Well, what's a close approximation? Well, what's the best you can show us in order that we can have an answer to this question that isn't just evolutionary philosophy?
So, I mean, you're pretty much looking at other species and comparing their Y chromosomes, the ones most related to us, primates. That's what evolution would predict differences stacked up on top of similarities.
Okay, wait, so is he in a position?
There's a lot of similarities there. The vast majority of the genome is similar. There's no problem for a couple of changes to stack up in the Y chromosome, especially when there's such a strong selective pressure for it to adapt, because other sperm are adapting, because the female reproductive system is adapting.
I'm not
sure what is insufficient about that answer.
Well, everything. It's just evolutionary storytelling. There's no model. There's no sophisticated mathematical model that you're showing. These changes that would have to take place, these are massive changes to the point where we don't have an analog. You can't show me two species, let's say even in the great apes or primates or the lesser apes, where we've seen today or in the distant observed past where Y chromosome differences can accumulate to that amount where you literally, okay, I'll ask you this question then. Why do humans have entire palindrome families? That's a specific sequence in the Y chromosome, and gene systems that are completely absent in pan, in chimpanzees and bonobos. That's what we see in the Y chromosomes of humans and apes. But if they're related, well, then the burden of proof is going to be on you, not to just, again, provide mechanisms. You got to model it out. You got to give us at least some kind of approximated ancestral Y chromosome sequence. And from there, we can say, okay, they've invoked sperm competition, they've invoked gene deletions, they've invoked duplications, so on and so forth, chromosomal rearrangements. Okay, now we can look at that ancestral sequence, do the math, map it out, look at fixation events, fixation rates. But so far, you guys aren't doing any of that. You're basically just saying, well, maybe this, maybe that, and then extrapolating it to say that, well, these massive differences must have accumulated because deletions happen, because chimpanzees are subject to greater sperm competition. These aren't answers, guys.
Why is it that you're focusing so hard on chromosome Y, but not on, say, like cytosome C, like where we have zero
Cytochrome
differences
C?
between, yeah, where we have zero differences between humans and chimps? I mean, we share 98.8%.
I feel like you're dodging now. You're trying to get off of the Y chromosome. If you want to do that, if you want to just concede, I already said in my opening statement in the rebuttal, it's the differences that make all the difference. Okay, we can both explain similarities. We can both explain some differences. But when you're looking at this kind, and I noticed you didn't even answer the question, why do humans have entire palindrome families and gene systems absent in pan? Can you just answer that? Just focus on this question. You guys failed on the previous question. Let's try to answer this one.
They diverged from a common ancestor, so it was
okay.
Easy
So you're
for them
assuming they diverged
to follow a mutational path that was different from one another and ended up in different results.
So which lineage lost or gained the palindrome families?
I don't know.
Well, I know you don't know. Nobody knows. Okay, what about the heterochromatin?
If humans and chimpanzees were exactly, it seems like you want humans and chimps to be exactly identical. And if
no, I don't.
Not,
I already said some differences are okay. It's massive differences we're dealing with here.
These aren't massive differences.
These aren't massive differences. Less than
30% difference in our DNA, and that's it.
No, it's the differences that make all the difference. You're ignoring the massive, even in the Y chromosome paper here. Notice this. They said
we're not ignoring the differences. 1.2% difference is exactly what we would expect to find after 7 million years of evolution.
Oh, so after 7 million years of evolution, notice this, right from your literature, their Y chromosomes differ just a little bit. No, enormously in size and gene content. Notice when you go to the visual here from 2024, take the human Y, bring it up to the chimpanzee Y, you're looking at 26.2%. You can't show us a Y chromosome ancestral sequence. You can't explain the palindrome differences. What about the heterochromatin? How do you explain the massive
the chromosome X? That's what, in the 98, the 80%?
Dude, are you even listening? It's the differences that make all the difference. Yes, similarities is agnostic on both sides. It's the massive. I've noticed again. That's 0 for 2 on questions. How do you address the heterochromatin? What happened to that?
The similarities are what prove that or demonstrate that we share a common ancestor with chimpanzees.
You mean
because the similarities can go either way. That's agnostic. We're looking at differentiating points, which you guys are dodging. What about this question, Austin? Where is the rate-based population genetic model that shows the duplications, the deletions, the recombination events, and the mutation rates required to produce? According to Spaghetti, there's barely any differences. Okay, let's look. New human-specific palindromes, restructured amplicons, loss of the entirety of the heterochromatic regions in pan. Okay, Austin, Spaghetti, give me an analog today where you can show me 2 lesser apes or 2 primates, okay, that have lost this much in their Y chromosomes. I'll accept an analog. Show me that, Austin. Do you have one?
Chimpanzees have had intense changes in their Y chromosome. Gorillas have had intense changes in their Y chromosome.
No.
Every primate species closely related to us has had big changes in their Y chromosome.
Chimpanzees and bonobos,
because they're promiscuous.
Okay, take those mechanisms and model it. Show me this. Show me I'll ask this. Does a model exist that actually answers this question where we don't just have storytelling and evolutionary philosophy, but we actually have a rate-based population genetic model, something empirical rather than, well, Sperm
Sounds like you're talking
competition.
about the Hardy-Weinberg equilibrium.
No, so what paper answers this question? Because I can tell you, 2017, they still admitted that sperm competition and faster rates of substitution, gene conversion to explain this goes untested. These are just ideas. These are guesses. But until you actually present a rate-based population genetic model, I'm wondering this, that the heterochromatin, okay? If we go to this visual for the audience, this one should be easy. This isn't easier. I get the ampliclonic palindrome families. It's difficult for you guys. Okay, what about this one? Here on the chimpanzee and bonobo, you'll notice the heterochromatin. It's completely missing. But on the human Y, okay, in gray here, it's present. Okay, heterochromatin is dense, tightly packed. DNA largely comprises satellite sequences. It's functional in chromosomal stability and gene organization. Anyways, so very clearly not a difficult challenge. Chimpanzees and bonobos, they're basically missing it all. Okay, why are they missing it all? If you're going to say, well, it was lost since the split. Okay, show me the numbers. Show me the model, and also why was it lost? Go ahead, Spaghetti. Let's have you answer this one.
Sure. Well, I like I said, I can't give you, and I've already conceded, I can't give you an exact map of what genes were lost in which year and which organism. What you're doing is you're saying the similarities don't matter whatsoever. They're completely agnostic. You're ignoring the 98.8% similarities in our DNA with chimps. You're taking a magnifying glass to the 1.2% and looking, you're saying, see, look, it's 1.2% different. Look at how different it is in these 1.2%. It's 100% different in these 1.2% differences. And you're completely ignoring all the similarities, not only the similarities, the identical placements of ERVs, which I'd love to go back to, which you think are magically placed there by God instead of being infiltrated into the DNA prior to a split, which makes way more sense. You say your interpretation of
okay,
ERVs
I get it. You're dodging the Y
is,
chromosome. I want to go one topic at a time, no slapping and running. We could go ERVs.
Yeah. Yeah, I think I have one that would hopefully be pretty quick. So, the topic of the debate, Donnie, is are humans and apes related? Would you take a couple of minutes or however long you think you need to take? Can you give us your definition of the terms human, ape, and related?
Well, specifically, it's are humans and chimpanzees related through common ancestry. I understand that James put the title "Are humans and the great apes related?" and that's fine because chimpanzees would be part of the great ape family. Taxonomically, you can look at different traits and features that would categorize, put us in the category of mammals, primates. You're going to look at certain features that put us into, like you mentioned, for example, the foramen magnum, mammal primate dentition. And so different morphological comparisons that would put us in the great ape family. But I also pointed to discontinuities, like in the fossil record of the Australopithecines, where we only have, Austin, one example of the hyoid bone in an Australopithecine, and it matches.
Wait. The question I asked was, how are you defining humans, apes, and related?
Yeah,
well,
let's start
related
with
would
human.
be.
Let's define a human.
Yeah, let's start with human.
Well, human. So a taxonomical definition of human?
What's your working definition of human?
A human would be somebody, well, like us, a human being that has the kinds of similarities where, like, for example, me and you would share more in terms of genetics, morphology, anatomy, certain anatomical or morphological features, certain genetic details that put us as Homo sapiens. But you want to bring in the chimpanzees because obviously there's going to be some differences, but less differences between us and a chimpanzee than us and, let's say, an orangutan or a fish. So taxonomically, I'm not sure what you're looking for in terms of a definition of human, but it doesn't answer the problems of your genetic level discontinuities that actually, because you asked, what does it mean to be related? Well, if we can actually trace humans and chimps back to a common ancestor, then I would say we're related, because you can trace me and my sibling, or me and my wife, or even me and you, back to a human common ancestor. But then you want to extrapolate that to say, well, we can also take humans and chimps back to a common ancestor, but that would be your burden in terms of relationship.
I think that there's actually a bit of a burden on you for that claim, because all we're saying is that evolution is the same now as it was in the past, and inheriting traits happens at the same rate now that it does in the past. And you're saying, no, there's a boundary in the sand. If you go back in time far enough, there's a hard line. And we're basically just asking, where's the line? What distinguishes a human from, say, like an ape? You mentioned there's a lot of morphological similarities between us. You might say, oh, we're both humans because we have two color-seeing eyes with binocular vision. Well, chimps also have that. Oh, well, we have hair. Chimps also have that. We have two nipples. Chimps have that. We have 2 arms, 2 legs with 10 toes, 10 fingers. All these things you can point to, chimpanzees have as well. And we can also look at, say, like behavioral patterns, social structures, and all the things that Austin mentioned. All these things humans and chimpanzees have in common. And the only thing that you can point to is these hyper fixated gene sequences.
No, again, you're misrepresenting me. I'm telling you that the overall similarities, okay, and homology, even your nested hierarchical patterns, are largely agnostic. You can explain similarities due to common descent. I can explain similarities due to common design, okay? Human engineers designing homologous patterns, why shouldn't God? And so we need to look to discriminating points, and that's where we could look to fossil record discontinuities. I mean, you guys brought up archaic humans. You mentioned the Australopithecines. Well, you guys love that topic so much, so why don't we engage a discontinuity that exists between humans and the Australopithecines, but we don't find these kinds of discontinuities within all of humans. So I can say, yeah, all humans are related. That doesn't require any kind of wild extrapolation fallacy. Okay, there's no major genetic level discontinuities. All our Y chromosomes are nearly the same, 99.9% the same. But once we start bringing in the chimpanzees, or even the Australopithecines, take Australopithecus afarensis, Australopithecus prometheus, right, Littlefoot. Okay, well, how do you explain those discontinuities if humans, Australopithecines, and the great apes are related, like the hyoid bone, for example?
I'm still not hearing a clear definition on what human is.
I told you.
What I'm getting is that when you try to define human, if you use the correct definition, then humans are absolutely apes. Everything that you would use to define a human also fits the definition of an ape.
Because I could look at a bunch of traits that make a vehicle a sedan, but that doesn't mean all sedans ranging from GM to Ford to Chrysler all go back to the same manufacturer. No, yeah, we can categorize things. The animals don't care how we classify them. The chimpanzees don't care that we classify them as pan and great apes. But again, similarities are agnostic. Overall taxonomy and classification systematics is agnostic. I'm arguing that it's the discontinuities in both fossil morphological data and genetics that is actually on the side of the creation position and is very troublesome to you on the evolutionist position. I'm providing you for the third time a discontinuity that answers your challenge, like the hyoid bone. And so, Austin, my question to you is this. If I could screen share, that would be great. We have ape-like hyoid bones. We have ape-like hyoid bones in Australopithecines and fully modern hyoids in humans and Neanderthals. So it actually turns out that the Neanderthal hyoid bone, which the hyoid is very important for what? For speech and articulation of language, okay? So my question is this. Where is the transitional form from Ardipithecus ramidus to Australopithecus afarensis, anamensis, so on and so forth, okay, up to Homo? Where's the transitional form that shows the gradual development of a human-style hyoid? So one characteristic that makes us humans is we can actually speak, and we're creative, and we can design some incredible things, okay? So the development of a human-style... So the development of a human-style hyoid capable of complex speech. What fossil evidence documents that intermediate stage? So in other words, Australopithecines have a hyoid bone that's very similar to the extant apes, not designed for speech. The human hyoid is distinct and unique from the Australopithecine hyoid and the great apes, but it's also similar to Neanderthals. So for that reason, we can make Neanderthals human. We also have their genes. Okay, so how do you explain, or where's that transitional series from an Australopithecine ape-like creature hyoid bone to the human hyoid bone, which is designed for speech? Go ahead.
One thing, if I can jump in really quick, Austin, one thing I'll just point out is that the car analogy that you use, Donny, really doesn't work at all when we're talking about evolution, because cars don't reproduce. And the reason that humans and Neanderthals share genes isn't because Neanderthals are humans. We're separate species. It's because there was interbreeding. So humans and Neanderthals interbred with each other, and then we have some percentage of Neanderthal DNA. But go ahead, Austin.
Well, I don't want you to slap and run. Cars don't reproduce, Ralph, but that's brand new to Matt, and I didn't know. I mean, thank God you got a PhD in what can and what doesn't.
I've heard you use that.
Adding reproduction. Listen,
you didn't know. I don't know. I'm just trying to help.
Well, no, and I appreciate it. It's very sweet of you. And so adding reproduction doesn't help your case. It actually adds more complexity, okay? We're finite, but God is infinite. So he was able to design the biological world with the ability to reproduce, to propagate, to pass on their genes, to vary, to adapt. We haven't gotten that far. Could you imagine we get to the point where a manufacturer can actually design a vehicle or a set of vehicles to self-replicate? They're going to save a ton of money. They're going to make $1 trillion, but it's also a straw man of what I'm saying. I'm saying perhaps we can get an idea into how God designed the biological world based on the way we design things. And it just so turns out that we as humans design in homologous patterns, in nested hierarchical patterns, which is why this whole point of similarity is largely agnostic, which is why I find it funny. I'm the one pressing you guys on the discontinuities, on the genetic differences, and you guys are dodging, dodge, duck, dip, dive, and dodge. I mean, we can hand it over.
The dissimilarities are agnostic in the same way that the similarities are, if you want to claim that, because there's nothing that could stop a God from designing organisms capable of evolving. And a lot of Christians don't see evolution and creationism as incompatible.
Well, it depends on what you mean by evolve.
You don't know.
Like, for example, the basic biological definition of evolution is what? Changes in allele frequencies, an allele being a genetic variant in populations over generations. So yes, we see that kind of horizontal change, but then you want to extrapolate that to mean, well, changes in allele frequencies means that fish can evolve into fishermen, or bacteria-like organisms can evolve into biologists. No, that's an extrapolation fallacy. No.
Changes in allele frequencies is the process that added up to all of the biodiversity on the planet.
Well, that's your assertion, your belief.
You're the one who's asserting that there's a line in the sand. We're saying
I've already shown you there's a line in the sand.
This process doesn't have an endpoint. You're the one who's saying a fish can't evolve into a fisherman, even though we know the process. We know that these changes can and do accumulate over time. And if you take something like a mountain, and you understand plate tectonics because you're not a flat earther, I think.
I'm not a flat earther. Oh, you
okay, good.
Not a flat, right? Yeah.
Yeah. So mountains form 1 inch at a time. So the plates smash into each other, and the mountain grows 1 inch, and then 1 inch, and 1 inch. And you're saying
that's the story.
There's no way that an entire mountain could form just from a few inches. You can get a few feet maybe of change, but you
can get it from rapid plate tectonics.
6 miles in the air.
You can get from rapid plate tectonics to mountain. But listen, we got limited time here. I don't want to go into plate tectonics. I really want an answer to the high. No, listen, I really want an answer to the hyoid bone discontinuity. I feel like you guys didn't answer the Y chromosome dissimilar. I want to say it again. It's the differences that make all the difference. You guys want to dodge all the major differences, the discontinuities. Just show me the transition.
Yeah, let me get one of you guys to respond to that. Then I'm going to let Matt jump in there in case he has something to say. I feel like I haven't heard enough from him.
All right.
But if either of you want to respond to the hyoid bone.
I know that I've looked this up before, and I'm pretty sure that we're getting either a slightly wrong explanation here, or there is an explanation. I can't remember.
What's the explanation? Okay, you can't remember.
Jed,
wait, I just want to say, because you're slapping and running. You're saying I'm giving wrong information, but you don't know why. You don't know. You know there's an explanation out there, but you don't know it.
I've looked this point up before, but I can't remember what
Yeah, because
I've
obviously
heard creationists make this argument before. If anybody wants to live search it, feel free to.
oh, okay,
Yeah,
so you're going to do
I'll
a
fully
Planet
admit
Appears.
that if I can just respond to that, I'll fully admit I don't know the hyoid bone evolution. I did do a quick Google search. What I found out is that Australopithecus afarensis has an archaic hyoid bone.
Yeah.
Homo heidelbergensis has a kind of a transitional or hybrid type hyoid
No.
bone, and then we see modern
Yeah.
humans having
No,
modern
Homo
hyoid
heidelbergensis and Homo neanderthalensis have their hyoid clustered within humans. That's why we know they were able to speak. That's why we know they're a sophisticated people group. So you guys are 0 for 2 on the discontinuities and the massive differences. Y chromosome, you admitted you don't have an answer to the heterochromatin. You don't have an answer to the palindrome and ampliconic sequences. On this issue, you have no answer for the transitional series from an Australopithecine hyoid bone. The only known Australopithecus hyoid bone that we have is designed and built like extant apes. Okay. Apes can't talk or can't speak like we do. Australopithecines couldn't speak like we do. There's enough variation in Australopithecines, like the ischium, for example, in the pelvies of the Australopithecines. Okay. So yeah, you can't show me or provide me an answer to this major discontinuity from a hyoid bone that's built like an ape, which does not allow for sophisticated speech and language, to the human hyoid. So that's a major discontinuity. And that's what I'm saying is it's these kinds of differences that make humans their own kind and that make the great apes and then extinct primates like the Australopithecines their own kind.
Yeah, I just want to make sure I understand Donny's argument.
All right, one at a time.
I was just wanting to make sure I understand the argument fully, is that we have hyoid bones. You go back in time to Homo heidelbergensis, they have hyoid bones, but then you go back far enough, and then you have more primitive hyoid bones.
No, it's not primitive. No. That's your evolutionary assumption.
Hyoid bones.
It's not primitive.
That's exactly what we'd expect with evolution. This
no, it's not.
You're looking at derivation.
Hey,
you're
one
looking
at a time. One at a time. I get I'm talking more, but you guys just made a point. I want to address it. Okay. No, you're saying primitive. When you guys use language, like when you're reading these papers in the paleoanthropological literature, and you see terms like basal or primitive or derived, this is evolutionary philosophy. Okay. The point is the Australopithecus hyoid bone. Notice this on the slide. The only known hyoid bone from Australopithecus afarensis, that's the Lucy species, is chimp-like, not human-like. You're going to say, well, that's primitive, or that it's chimp, as in it looks like is the chimp hyoid bone primitive? No, they exist today. That's the way it's designed. It has the bulla and air sac features of apes, not the slender speech-adapted structure of humans. That's not a transitional trait. That's a categorical distinction. The fossil record still lacks any hyoid. Look it up. Google it right now. Ask ChatGPT, Austin, showing a stepwise evolution toward human speech anatomy. What makes a human? Well, one trait or one feature is the fact that we have the ability to articulate creative thoughts. We have the ability to speak, for example. We have the ability to have a spiritual connection to God, but we don't see this in the Australopithecines. We also don't see this in your apes today.
You do a lot of question begging with references to God, but so you're claiming that every hominid fossil that doesn't have the hyoid bone is a chimpanzee or some sort of ape. So if you look back at, say, like Homo erectus, which I don't know if that's a hyoid, I'm assuming it doesn't have the same hyoid bone as we do. You're going to say that a Homo erectus is an ape and not a human. Is that right?
Homo erectus is a human. I could show you articles that talk about how Homo erectus is sailor, very sophisticated people group. For the postcranial material is 100% human. That's obvious. There's some environmental morphological variation in the skull, for example. The teeth, it's a little bit more extended in the snout region, prognathism, all these features that because we know environment can oftentimes dictate morphology. And it's interesting because it's not even you guys, I've seen you, Spaghetti, I appreciate your knowledge on the Australopithecines. I watched your debate with Jim Bob, but we understand, I would agree with you that the Australopithecines had a form of bipedalism, okay? They had a mix, actually, of arboreal and terrestrial traits similar to gibbons. But even in their pelvies, which their pelvis actually looks more similar to the human pelvis. Notice here, you got the chimpanzee pelvis, which is longer, right? Then you got your Australopithecus africanus. That's a different species of Australopithecine I'm screen sharing right now. And humans. So you can see their pelvies are very similar. Short and wider. The Australopithecines have that iliac flare. Okay. But
It looks like a gradual transition to me.
well,
yeah,
no,
exactly.
but here's the thing: is it a gradual transition, or can we point to discontinuities that you can't explain, like the hyoid bone,
You
or
have a
even in
major
the
ontological issue. You're just calling derivation discontinuity. You're
okay?
treating any difference as if it
No,
substantiates a discontinuity,
it has to be a big enough
which it
difference
doesn't.
that creates
You
a
would
gap.
expect differences stacked up on top of fundamental similarities. You're saying ignore the foramen magnum, ignore it migrating up to the front of the skull, ignore
But
all of the postural changes with
you're
the valgus
asserting it
knee,
migrated. We don't
ignore
see that.
all of those morphologies, and just pay attention to this hyoid bone thing.
No,
It's cherry
I'm gonna
picking.
point to another in the pelvis and gait. Okay, so in the Australopithecines, you got laterally flared ilia and long ischium. Okay, which shows that they were walking upright, but in a side swaying, an inefficient kind of locomotion where it's like a bent knee, bent hip kind of walk. Anyways, my question to you is this: we know, okay, as far as my research has suggested, I'm open to being corrected on this one. Hyoid bone for sure. That's it. You guys can't explain that one. This one's an interesting one. The ischium. The ischium in your Australopithecines is longer in the pelvis. Okay. But guess what? Your extant apes like chimpanzees, bonobos, gorillas, and orangutans, they have the longer ischium. Humans have a short ischium, interestingly. And that is why we have a more efficient form of bipedalism, upright walking. Okay. But amazingly, it turns out that Homo naledi, who we would say are humans, and Homo floresiensis, that's Hobbit man, also had a shorter ischium. Okay. So my question to you, Austin or Spaghetti, show me any Australopithecine or extinct ape that had the short ischium like modern humans today. Go ahead.
Are you done with the screen share?
Yes.
All right.
And then we can throw it to Matt. You guys answer that question.
Yeah,
about
Matt's
the
been
ischium,
waiting for a minute.
then we'll throw it to Matt.
We're about halfway through the open discussion, just a little over. We got about 27 minutes left-ish, maybe a little less.
So Spaghetti.
So yeah, you guys go ahead whenever you're ready.
Spaghetti. Australopithecines, because we got to take a holistic approach. We're not just taking cherry picking one or two features. Let's look at the cranial. Let's look at the postcranial material.
Sure.
Let's look at the genetics if we have it. So answer the question about the long versus short ischium. Go ahead.
Yeah, I'll answer the question about the ischium in just a second. I did want to go back to the hyoid bone just for a second. I did a quick Google, and I found a study from 2008 talking about Homo erectus. I just want to read a quick excerpt. There's a total absence of muscular impressions on the body's ventral surface, which suggests a reduced capability of elevating the hyoid bone and regulating speech. So, in other words, the Homo erectus had a reduced capacity of speech compared to modern humans. So this could serve as a bit of a transitional form, if you will, for hyoid bone evolution, where Homo erectus had less speech capacity as modern humans, more so than an Australopithecus. So it's somewhere in the middle. So for you to claim that a Homo erectus is fully human is just patently absurd.
It is fully human, but there's evidence for environmental degeneration in so-called hominins like Erectus, Naledi, Flores. What inbreeding, isolation, what those things do is result in genetic degeneration. Inbreeding reveals the hidden. Wait, I know you're excited, but just a sec. Reveals the hidden reservoir of genetic mistakes, and that could actually show up in the morphology. And so notice how you said reduced ability to speak, but not the non-existent ability to speak like we see in extant apes and also the extinct Australopithecines. Yes, Erectus, being a little bit degenerate due to the inbreeding and isolation and environment that it existed in, is very similar to what we see in Naledi and Hobbit, where that affected its brain, because brains are energetically costly. That's why you oftentimes see reduced brain size on island populations like Hobbit, because they're trying to preserve resources and energy. Okay, what that results is a diminished form of speech. Yeah, that's adaptive degeneration, but not the complete lack of ability to speech. You extrapolate that to mean, well, this is transitionary, even though it could be due to pathology inbreeding, isolation, and degeneration.
So
Anyways,
your
the
claim,
ischium, you wanted to answer
your
the
claim is
ischium?
that the lack of ability to speak as well as humans in Homo erectus is not an evolutionary step from less speech to more speech, but instead it's from more speech to less speech because of inbreeding. And as soon as you mention inbreeding, the first thing I think of is Noah's Ark, when humans were reduced to a population of 8 individuals.
Yeah, but that's not the topic.
Crazy.
But here's what's interesting. Okay, there's two different starting points: creation and evolution. Spaghetti. The evolutionist explains the vast majority, if not all, DNA variety as being the result of what? Mutations over time. Creationists, we explain the vast majority of autosomal biparentally inherited nuclear DNA as being the result of created heterozygosity or design diversity, which means at the flood, at the ark, firstly, it was a single generation bottleneck followed up by rapid and exponential population growth. There wouldn't have been enough mutations accumulated by that point in time for all of these mutations in recessive form to come to the forefront and actually lead to rapid genetic degeneration. It'd be a problem for you in the out of Africa model.
Yeah, well, I want to correct something you said, if I may. So you said that we have two starting points, creationism and evolution, but that's not true. Evolution is not a starting point. Evolution is the product of observing the evidence and following where it leads. Creationism is a starting point because creationists start with their conclusion already in mind and then work backwards, cherry picking evidence which supports their preconceived notions.
Well, that's your assertion. It is a starting point, and I've already showed you that it's you that's accommodating the evidence. The audience can go back and see you guys completely failed, dodge, duck, dip, dive, and dodge, the 5 Ds of evolutionist dodgeball, answering all of my questions associated with the Y chromosome. You didn't answer the hyoid transition. You still haven't answered the ischium. I just explained to you how, from the creation starting point, inbreeding is not a problem. We can bring in mitochondrial DNA haplogroups. It turns out, amazingly, that there's 3 main major haplogroups, L, M, and N, in the mitochondrial DNA, all worldwide mitochondrial DNA, Spaghetti. And I could show you a phylogenetic tree of this, but I won't for the sake of time. It goes right back to those 3 main starting points. Well, guess what? Off of the ark, you have Noah, his wife, their 3 sons, Shem, Ham, and Japheth, and their 3 wives, Noah's daughters-in-law. And wait a minute, 3 major haplogroups, L, M, and N. Is that coincidental? How do you address that, Spaghetti?
Yeah, the topic for today's debate is not.
So you bring up off-topic points, and now because you can't answer that, run away.
You can make up whatever nonsense you want about whatever.
You're saying the fact that there's 3 haplogroups.
You guys lost. It's over. It's another easy win.
What a stretch.
When you appeal to magic, anything is possible.
All right,
You guys
That's a real big reach.
The Y chromosome dissimilarity, the hyoid bone, the ischium.
Let's let Spaghetti respond, please. Let him respond, please.
I think I answered the hyoid bone with to satisfaction. I mean, I stated how Australopithecus had a reduced size, like you said, similar to extant apes. Homo erectus had kind of a transitional hyoid bone, and then we see normal hyoid bones in modern humans. Regarding the ischium, I can't give you exact, this fossil had this ischium and this one had that ischium. One thing I can say is that evolution isn't necessarily a completely continuous lineage, right? So you're going to get changes, traits are going to come and go, ischiums might get longer and shorter. And then when we look at these hominid fossils, it's also not a direct ladder. So we don't see a 100%. You're familiar with the march of progress, and I'm sure you've heard that that's not an accurate representation of evolution, but instead it's a tree. So we have lots of extinct hominids that just died off, right? So we have ischiums that have gotten longer, then that lineage died off, and we have ischiums that got shorter. Maybe that lineage died off.
This is all storytelling and evolutionary philosophy. You haven't answered a single, maybe this, maybe the ischium. I'm asking a very clear question, Spaghetti. You've dodged the entire time. Then you're the one that goes to the Bible. I completely dismantle you. Okay, I give you a scientific spanking on that issue, and then you're the one who says, well, that's off topic. Yeah, because you don't have an answer to the 3 major mitochondrial DNA haplogroups. You did not answer my challenge on the ischium. Okay, my question was this:
3
show me a single. Listen, I know you're excited, Austin. I got to reiterate my question because Spaghetti dodged.
These don't equal each other.
We're going to throw it to you. I'm going to reiterate my question that Spaghetti dodged. The ischium. Show me. We have Australopithecine and extant apes. They have the longer ischium, which shows us that they walked upright, but in more of kind of a clumsy or less efficient way. Bent knee, bent hip, okay? Humans have the short ischium. So show me an Australopithecus or an extant ape or extinct ape that had the short ischium.
Say we don't have one. Say we don't have any fossils. Could it still be possible that humans and chimps are related, even if we didn't have a single fossil showing it?
No, because we're taking a holistic approach. Okay, we're looking at the genetic data like the Y chromosome.
What you're doing is you are
what
pointing
you're doing
out,
is not answering any
you're
question.
pointing out a piece of ignorance, a piece of data that we don't have.
How many points of
and
how many
pretending
pieces of
as if
data
that
do
is
you need?
Let me just try to
disconfirming
clarify
data,
this real quick.
which is not the same thing.
Austin, are you saying is it logically possible? Is that what you're asking?
Sure.
Okay. And
Yeah, it seems like, Austin, you're also pointing out that you're also pointing out that Donny is using the argument.
I just want to go on record
ignorance
for the
fallacy.
audience. You guys have no answer to the iskim, right?
Basically.
Because so basically you have no answer to the iskim. Okay, Matt, let's get you in on this. I've been cross-examining these guys into oblivion for the last 30
I'm not a
minutes or so.
paleontologist.
Well, you're not a geneticist either because you didn't have answers to the Y chromosome dissimilarity challenge either. So what are you?
No, I'm not a geneticist either.
Okay, Matt.
And neither are you, Donny. You don't have answers to endogenous retroviruses.
Yes, I
All
do.
you can do is claim
retroviruses.
it's magic, and God put the ERVs in the same exact spots in human and chimp genomes. What a coincidence that God just happened to put all these ERVs in the exact same spot. This is a clear and probably the biggest demonstration of evolution.
Have you read my book on endogenous retroviruses, Spaghetti? Because I'm about to slaughter you. Have you read my book?
I have not read any of your books.
So you're about to get slaughtered.
To.
I'm going to ask a very basic question right now. Show me one paper. Show me one. I want you to pull it up. We've been doing all the screen sharing.
I think
we got the receipt.
Yeah, I'm gonna have to call that. I think Matt is sharing his.
Yeah, but I don't want him to slap and run. I don't want to. I want to ask him a single question on ERVs, Mike, because we're not going to let him slap and run. Okay? He challenged me on ERVs. I got a single question. He can answer it.
Make it brief because I do want to be fair to Matt. He's here.
Okay.
It's everybody's time.
Okay, Spaghetti. Show me a single paper that demonstrates a non-functional endogenous retrovirus going from non-functional to something incredibly functional in embryological development. I showed you a paper from the conventional literature that literally said ERVs are functional in embryological development to the point where if we didn't have them, we wouldn't exist. Show me that paper right now.
I mean, I can't pull out a paper in front of me, but I'm not sure what your point even is. ERVs.
My point is the better explanation for ERVs is special creation
course.
because of their essential functional roles. So, do you have a paper
There's
that shows an
maybe
ERV?
5 endogenized gene sequences that have a role. That's less than 0.00001%.
The fact that the ERVs
my source on this is Lawrence Moran. I trust him way more than you.
We have evidence for over 80% of the genome as being active and functional. Within that 80%, we have ERVs. We understand. I showed you a dozen papers where ERVs are functional in embryological
Donny, this
development.
Is completely irrelevant.
Even if 100, the immune system.
Even if 100%,
I am showing you why you can't account for these essential functional roles in ERVs, which is why the creation position has the superior explanation. And Spaghetti, you're 0 for 5. Matt,
this is completely irrelevant.
We just explained it to you, Donny. There's 5 endogenized gene sequences, maybe 5 max out of hundreds of thousands. It's less than 0.0001%. Sorry, go ahead.
I'll also point out that even if 100% of ERVs were repurposed to have a function in modern humans, that doesn't prove that they didn't get there by evolution. It doesn't matter if they have a function or not. The fact that they're there and exist in the exact same spot in human and chimp genomes demonstrates that they got there before the split happened. What a coincidence that this retrovirus just happened to come into the exact same spot in human genomes and chimp genomes independently. I mean, what are the odds?
Are we going to talk about ERVs or are we bringing Matt into it? Because Mike, I can go for another hour on ERVs.
No one's stopping Matt from hopping in.
We have 5 minutes on the open dialogue left, so let's go ahead and wrap up with
I'm going to answer their questions because they had a few that were jumping around and we missed them even in the opening. So I'm going to jump backwards in time and we'll get back to ERVs.
All right.
So I thought this was pretty funny.
If I could just ask Donny, try not to take this anywhere else. Just let Matt do his thing, please.
This was pretty funny. I found this in Google Dictionary, what a human is. I just thought I would throw that up there because that's pretty funny. Human, person distinguished from an animal. In science fiction, an alien. Anyway, when you were to see a fox Do you think that's a primitive skull when you look at modern day dogs today? Nah, you would never say that they're primitive. We just see phenotypic change. Shifts are relatively fast. So when we look and we want to know, is there discontinuity there? This is baraminology. The same thing exists in the evolutionary literature. You can see that there's groups. All they did different was put a line in between them. That's really the only difference. But when you look at an early human, like this would be considered the earliest Homo sapien, as of recently, that just changed, but we'll get to that. The Jezebel eyehound skull, when you line it up with a Homo erectus, it actually overlaps almost identically. I didn't drag that picture in here, unfortunately. So I'm gonna have to skip that. But right now, they found a skull recently, dated 1 million years old. And when we move that skull directly over the exact same early Homo sapien and Homo erectus skull, the same exact thing. The pattern matches identically. So that's why in fossils, we set a boundary, a genetic boundary, and a homology boundary as well. So that's how we do it with when we don't have any genetics whatsoever. But you brought up a question about cytochrome C. Okay, well, guess what? Let's just look at cytochrome C then. They noticed in 1978, that's what this study was, that the chicken appears to be more closely related to the penguin than to the duck. And the pigeons, the turtle, and a reptile appears more related closely to birds than rattlesnakes. And man and monkeys diverged from the mammals before the marsupial kangaroo separated from the placental mammals. Here we see that the horse is actually less related to us than a kangaroo. So even the phylogeny there breaks down. And Eugene, I don't know what his last name is, sorry, I'll butcher it, 2014 noticed that cytochrome C of the American alligator was more similar to us than a primate of a monkey. So even the most conserved region, the best one to build a phylogenetic tree, breaks down after not so long, going just backwards in time. So then when you compare it to cytochrome B, it completely contradicts it. It's absurd phylogeny of mammals. And here's a little chart. It nests whales and tarsiers with cats and humans all together in the same one. That's why the scientists are saying we have no evidence at all that a tree of life is a reality. The tree of life
Matt, I appreciate that you've been being so patient and waiting for your chance to talk. Can I ask one quick question?
Yes.
Do you have the actual numbers of the amino acid differences in the comparisons you're making?
The kangaroo was 10 differences, so it was much closer by, and then the horse was 2 differences further away. The exact number in this one, I just grabbed their quotes. I did not grab their chart, but I do have a chart. I just took a screenshot, but it looks horrible.
If I could get that info after the debate, I would appreciate it.
Okay. So the next one, there is no theory holding that molecular similarity necessarily implies an evolutionary relationship, and molecular data that contradicts it's usually dismissed. In the evolutionary college books, Tree Thinking and Evolution 1-on-1, they don't even dare to name a single tree root common ancestor. But when they go back in time and they look in a fossil record for one of these bottlenecks, they said there's no trace in the geological record of any global event 200,000 years ago, but yet that was the massive bottleneck. So when people say, well, it's only religious people that believes in these global floods and things like that, actually, no, this is a book right here, Up in Earth and Upheaval, written by an atheist, based on a global flood. He talks about how he discovered whales were on mountains, but whales to move up onto a mountain through the slow geological process of 1 inch a year, for example, whales can't be on a mountain because they're a new species about 55 million years old. Endogenous retroviruses were actually predicted in 2000, back when they were actually supposedly only junk DNA with 99%, like literally they were functionless. Go. I see a question.
Yeah, can we slow down? Can we back up a little bit? Back up to the slide before. So let me try this. Let me try and steelman your case a little bit. So the case is that what, 6 to 12,000 years ago or 4,400 years ago, however many years ago, all the species got on a boat, but we can't fit 8.7 million species. So it wasn't all of those species. It was progenitor species that diversified into a kind after the boat landed. So let's say that, hypothetically, that I'm ready to change sides. Are we saying then, and I just have some questions, are we saying then that all of this taxonomic development and change, all of this geological, volcanic, meteorological, all of this change happened in that like 4,400 something year period?
Most of the sediments would be heavily distorted during that time. Correct. That's in the catastrophic plate tectonics. Mountains were about 5,000 feet high, and then they pushed up into their massive heights that they're in today. Yes,
So,
and then
so
the
yes,
species.
there was. You're saying
Austin,
yes, there was
Austin.
immense, massive change in taxonomic phylogenies in the array of species. It did happen over the 4,400 years. There was big change
I'll let you
in the
answer
array of
that, Matt.
species.
I will let you answer that. I just wanted to let everyone know we just hit the 50-minute mark for the open dialogue. I'll give you guys 5 more minutes since Matt didn't get a ton of time to
Sweet,
talk.
thank you.
Okay.
And do we have 5-minute closings, Mike?
Yeah, after this 5 minutes, you'll each get a back and forth of 5 minutes each. I think that breaks down to 2.5-minute closings apiece, but if you guys are all not very strapped for time, I'll let it kind of take a little bit.
Okay. Yeah, so we're going to say that the species that were on the ark were the paternal ancestors. So there were 2 wolves on the ark. They would have got off and diversified, and then today, because of breeding, of course, we get all the dogs. And so, yes, we would say that it happened rapidly since the Earth was empty. So we got to say, yeah, we want to know. So you believe the same as us because of the animals' descent with modification. It's the same thing
So-
rather than ascent.
So my follow-up question would be then, what do you think the array of species is going to look like in like 500,000 years, in 5 million years? Do you think it will include entire new clades of species that currently don't exist?
I think what would happen is if picture Paris Hilton and she brings her little chihuahua on her airplanes with her, and all of her friends do the same thing, and there's a global bottleneck, and all we're left with are those little chihuahuas to breed off. That bottleneck wiped everything out. That chihuahua now becomes the paternal ancestor to all life. Is that going to have less genetic diversity than the wolf did, or can we create all of the species of dogs today from those? Now it's going to get less over time. Every bottleneck is going to reduce genetic diversity over and over and over, because there'll be more bottlenecks over 5 million years of time. We've already had 5 global, but we're going to keep reducing over and over.
So yes, there will be new clades of organisms that don't currently exist, but because of genetic degradation, not evolution.
What do you mean by clade exactly? But like a new clade of dogs, or from that, we're going
I
to
mean,
be
like, you have the clade of ungulate quadrupedal land mammals, and then within that clade is another clade of aquatic land mammals, the cetaceans. Is there going to be changes like that?
No, it'll be more limited. What we can do with breeding. Breeding exposes the limits of what can be related. We can crossbreed and hybridize different things, and then that's like we have as fast as race horses that we can get. We drop millions of dollars into horses. We can breed them in anything. Cattle too. We want them to get big as possible so we can eat them. Well, if we could make cattle crazy looking, hybrid with other animals like an elephant, we would. But there seems to be genetic boundaries there. So we're just going to say, just keep following those boundaries and limits that we find, and it'll just keep getting reduced over time from every bottleneck.
If I can
I
hop in here for
appreciate
a
your
second.
candid answers so much. I'll wrap up on this. The reason I asked the question is because it causes me a lot of cognitive dissonance to try and say all of this diversification, the development of new clades, all this change could happen from the arc to now, but 500,000 years into the future, there won't be these big changes, or that's the first point. And the second point is, I understand that your argument involves genetic limits. I would need more substantial evidence that the genetic limits exist, but I appreciate your candid answers very much. Thank you.
Okay.
Matt, if
And
I can
fun
ask a
spaghetti.
question. You mentioned that foxes are a separate kind from wolves and dogs. So how do you make that distinction? What makes you so sure about that?
I went to the DNA barcoding website where they test something called the cytochrome C, which is in the mtDNA, and it's highly conserved across everything. And because it's so dirt cheap to test, they do it on everything. So they've tested millions of species. So we have all the different dogs tested, all the different wolves tested. So I put everything together, and I was like, okay, if wolves and dogs, I'm going to run all dogs and make and put it into a AI and say, make a consensus sequence out of that after the 650 base pairs. Then do the same thing with wolves. You do the same thing with foxes. And then you take that consensus sequence and you go backwards in time. Does it converge with a common ancestor? The dogs did with the wolf, but the fox did not. There were 12 fixed differences between them or more. So they could not have been on the ark, or they had to be on the ark together, or the last bottleneck, if you would say. They were not converged to be the exact same kind.
I guess I'm a little confused because you're comparing modern dogs to modern wolves, as if modern wolves are the ancestors of modern dogs, but modern wolves share a common ancestor with dogs, but they're not the ancestor of dogs.
That's correct.
In the same way that humans share common ancestor with chimpanzees, but we didn't evolve from chimpanzees. We both trace back to a common ancestor from the evolutionary point of view, of course.
All right, one quick response, if you will, and then we're going to get into the 5-minute closings. Cool. Now is a great time for people to super chat a question for the guests and the debaters tonight. So we got a couple of the super chats stacked up in the background here, so they're all ready. Just get the last couple in, and we're gonna bang them out here in a second. So
yeah, send in some super chats on cytochrome C, ERVs, pseudogenes. Let's have some fun elaborating even more on these technical topics.
Sure. I would just answer with an evolutionary genome sequencing of its kind, saying that all dogs descend to a single wolf, disfavoring models which dog lineages arise separately from a wolf population. So the ancestor to all dogs had to come from a wolf that we would say was on the ark, that you would say came out of the last bottleneck. So there was a wolf ancestor, no doubt. And so we would say we can test that because going backwards in time, that consensus sequence converge. We can't converge a human and a primate missing link common ancestor going backwards in time. And that's what we did with the DNA barcoding, right? You take that, the sequences go backwards in time, but do they converge? We don't see any convergence. Matter of fact, there's 65 different base pairs different. That's massive. So there's no convergence. So there's a separate line of ancestry. That's in genetics. Then we find it in homology. Then we find it in taxonomy. That's why it's called the taxonomic boundary paradox. And so there's separations of boundaries in every single field. And that's what creation is predicted. So that's what makes our model so strong. We predict these things. And then, so yeah, sure, we'll start with a idea. Yeah, God did this all 6,000 years ago. Sure, we can say that all day long, and we're starting with the answer, and then we're trying to retrofit. Or are we actually making predictions based on that, and then finding those predictions? That's what we've done. And if the mutation rates didn't land on the biblical timeline, we're done. We're finished. It's over. But they all do? I mean, what are the odds? All right, your turn.
All right, I think this is where we'll go into the 5-minute closers here. So I appreciate y'all's time and cordialness on the discussion. There was only a couple times where I felt like we got a little out of bounds, so that's great. All right, so we'll start the 5 minutes, and I believe, Austin, you guys opened, right?
Yep.
So then I think y'all are going to be closing it.
Cool.
All right, and starting now, go ahead, Donny.
Oh, so are we the negative side starting closings first, or because they started opening? So do they go first?
I think who starts it closes it.
Okay, yeah, we can do that.
I mean, if someone knows better than me right now in the chat, let me know.
Oh, no, I'm happy with that.
I think that's usually how it goes.
Go ahead, Donny.
Okay, let me get my notes up here then, and good fast-paced discussion. I'll
start the time when you officially start.
Yeah, let me get my things in order here. We discussed a lot, so I want to make sure.
Let me remind the audience, DebateCon 7 is going to be next February in San Antonio in my backyard. I live here in Central Texas, so you guys, everybody should come to that one for sure. It's going to be bigger and better than ever, so come meet me there. I'm hearing that my volume's low. Let me check that real quick. Are you sharing the screen? Let me.
Yes. Can you see the screen?
Okay. Just give me a heads up so that it doesn't throw it off before I got it ready. All right, we're good.
Okay, here we go. I'll start the timer now.
Transition.
5 minutes. Matt, I'll take about 3. You take the last 2. Okay, well, that was a lot of fun. The burden of proof was on them. Are humans and chimps, or humans and the great apes related through common ancestry? It's the differences that make all the difference. And I challenge them on many major differences and also discontinuities in the fossil record data with the Australopithecines, for example. And so what we got was a lot of dodge, duck, dip, dive, and dodge. We saw that recently last month with our previous interlocutors. Y chromosome dissimilarity, not answered. Again, I predicted all they would do is give just so stories, evolutionary philosophy, mechanisms, but mechanisms without a model, mechanisms without a reconstructed Y chromosome ancestral sequence means nothing. It's not empirical. All we got, again, was a bunch of storytelling. They did not meet their burden of proof. Hyoid bone transition. We got the Australopithecines with a hyoid bone not designed for sophisticated language and speech, similar to extant apes. And the human hyoid is unique. Discontinuity there. The iskim, of course, they didn't even try. LMN haplogroups, mitochondrial DNA worldwide goes back to 3 major haplogroups, which fits the biblical model beautifully. And functional ERVs, of course, they could not show us a paper that demonstrates the origin of these incredible functional roles with ERVs. So clearly, if they had read my book, I've got a list of all of Austin Sims challenges and questions here. Perhaps Austin Sims and I can have a good informal discussion sometime after this on just ERVs. I'm happy to answer all his questions. Viral codon bias, for example. Well, codon bias reflects the host the virus came from, not the other way around. Retroviruses match host codon usage because they ultimately derive from host genome machinery. So here's the ultimate question. It's a matter of origins, okay? Maybe they can answer this in their closing statement, which means they'd have answered at least one question tonight. So what came first, the retrovirus or the host? Because a retrovirus requires a host to replicate in the first place, okay? We as creationists, we argue that God would have front-loaded the original created kinds with various classes of functional retro elements or retrotransposons, and due to mutation accumulation, due to degeneration after the fall, you'd get directly from what many in the conventional literature describe the escapee hypothesis, you'd actually get exogenous retroviruses escaping, leaving host genomes to infect other hosts. And that's how we would explain the distinction between one of Austin Sims' challenges, the differentiating fixed versus unfixed ERVs. The koala bears, for example, have unfixed ERVs that the functional fixed ERVs are actually destroying. So I don't know if we got an answer to this. I don't think so. What is the evolutionary pathway by which an LTR evolved? How did the preloaded regulatory functions inside ERVs arise in the first place? Why do evolutionists claim vertebrates borrowed regulatory functionality from viruses, but never explain how the viruses evolved that regulatory functionality in the first place? How does random viral insertion produce coordinated development timing? Anyways, we addressed it all. Chromosome 2 fusion, hierarchical patterns in the cytochrome C. The Phoenix virus experiment is exactly what we would expect if what I just iterated was true, the escapee hypothesis. And so that's basically it. I mean, we addressed every single point they brought up.
So you're at 3 minutes and 30 seconds if we want to hit it over to Matt real quick.
Yeah, let me just finish my thought here. And so we addressed all their challenges and questions, and they addressed nothing. Matt, over to you, brother. You got a minute and a half, roughly.
You going to share your screen,
Okay.
Matt?
Yeah, please.
All right.
Minute and a half. Okay. The observed mutation rates that I went over at the beginning always disagree with evolutionary phylogenetic mutation rates that are based on radiometric dating for the fossils. That's the whole question of ancestry, right? So if we can explain everything within the short time period and mutation rates confirm that, and it's not just in humans, it's in all life, all animals tested. You go back in time very recently to a global bottleneck. There is no global bottleneck in the evolutionary model. That completely destroys everything. When you're trying to look at the diversity of life and go, how old is that species? And it shows it's young. That precludes evolution from happening, especially when it never lands on an evolutionary predictive date. So to wrap up, I guess I'll just say they mentioned earlier that there's almost no differences between chimpanzees or primates and chimpanzees and humans. Let's look at some of them. There's an arch in a human foot. It's not found in chimpanzees. A subcutaneous layer of fat that runs through our body keeps us warm. No primate has that. Goosebumps. We get those. Primates do not. They might have hair, but they sure don't grow up very long. How about beards? We apparently evolved one from being punched in the face. Body fur, sure, they have some, but very, very different. So much so that when there's a forensic analysis, they can tell you if there was a primate in the house or a human. The melanin, we have melanin. Primates do not have melanin. Only humans can blush. There's contrasting color patterns seen with our eyes. The sclera is very different. Blood. If you got a blood transfusion from a chimp, you would die. But yet we can almost take one from a pig. That means there is much more similar. We have rose hip neurons in our brains. Primates do not. We have completely opposite digestive tracts. Apparently, we decided to evolve one, even though we were alongside of them eating the same foods almost at the same time up until modern recent history.
30 more seconds, Matt.
Okay. This means that humans needed to evolve entirely backwards the digestive tract based on the foods they were eating. Their appendix, completely opposite. So many differences.
Awesome. Thank you for that. And there's 5 minutes left for super chats before the Q&A. I'm just reminding everybody, get ready to ask these guys whatever you want. And we'll kick it over, starting the timer for the other team. Go ahead and you're ready. Do any of you guys need to share your screen or anything?
No, that's okay. I can go first if you want, Austin. So,
Yeah, you're cool.
let's see. The rate of mutations does not directly affect the rate of change. The environment is the driving force of the rate of change and not necessarily the rate of mutations. Donny claims his interpretation that ERVs were magically placed in the genome by God is on equal footing to ours, that ERVs infiltrated the genomes of both humans and chimps before our split. It's completely ridiculous. The body repurposing ERVs to serve a function does not debunk evolution. Even if every single ERV in our entire body served a function, that wouldn't debunk evolution at all. The notion that an omnipowerful God would need to infect his perfect creations with viruses in order to function properly, instead of just making their own DNA carry out the necessary functions, is laughably absurd. This isn't really on topic necessarily, but the Bible does say that the earth is flat, and Donny confirmed this by citing Isaiah 40, which calls the earth a circle, or in other transitions a disc. Matthew says that you can see every kingdom on earth from one tall mountain. Samuel and Solomon Job all claim that the earth rests on pillars. It's laughable that they think Noah's Ark, creation, and the Tower of Babel are actual history. Donny complained when I initially brought up Noah's Ark in the Bible, but then he throws a fit when I try to get us back on track. Noah's Ark doesn't explain anything. If anything, I was trying to save Donny a little bit of grief, because it creates a billion different problems for creationists. How did the animals evolve so fast afterwards? And then why did evolution slow down immediately? How did Noah store the necessary food to feed those millions of animals? Where did the water come from? Where did the water go? What about the heat problem caused by creating giant mountain ranges in less than a year? This heat would be more than enough to obliterate the ark into smithereens multiple times over. Why are all extinct fossils neatly placed in rock layers corresponding to evolution instead of scattered randomly, like we would expect if there was a chaotic flood? The Bible does not correspond to observed facts about reality. Human fossils cannot be found in rock layers older than 300,000 years. Instead, we see a progressive lineage of hominid fossils. Throughout this lineage, we see a progression of flatter faces, reduced canine teeth, bigger brain cavities. Donny mentioned how the Australopithecus ischium's differences debunks evolution. I also already mentioned evolution is not completely linear. Some phenotypic traits come and go. Evolution is a tree. No one is ignoring differences between humans and chimpanzees. The differences are what make us distinct species. The similarities demonstrate our relationship, which is why our creationist opponents insist on ignoring them and claiming they don't matter. Donny hyperfixates on specific examples, and even when we produce sufficient answers, like with the hyoid bone, he's still unsatisfied. And I'll kick it over to Austin.
All right, you got. I'll give you another 3 minutes, even, just to be fair.
Thank you. So I love Donnie's passion, but I have to opine still that for many of the reasons Jed stated too, that debating creationism is kind of fish in a barrel. I think Donnie kind of struggled with even just the basic definitions of what evolution is, what a human is, what an ape is. And when he uses the correct definitions, he's pretty much conceded the debate, which is why he's got to create his own idiosyncratic definitions and insist everybody use them instead of the ones that actually make sense. If there were any ERVs in your genome that were completely functional, you wouldn't even exist. They would still be producing the viral product instead of creating the cells that create you. And what is partially the problem here is that I think our opponents, one of the idiosyncratic things they do is they change the definition of scientific epistemology, of scientific evidence. We're not saying that we're gathering proof. We have evidence. We have a possible, falsifiable, quantifiable solution to unanswered questions about unsolved problems in our attempts to better understand the universe and natural history using what we learn to improve human existence. We're not saying that evolution won't change when we get more information. It very well might, but currently the main proportions of the model, cumulative, random mutations being compounded by natural selection, that's highly unlikely to change. We had 5 lines of evidence: morphology, genetic similarity, chromosomal fusion 2, cytochrome C, ERVs. All of these are different lines of evidence, all fit each other and support the same conclusion. The rebuttal needed to be overwhelming, and then we didn't even talk about any of these points really all that much. We talked about ERVs a bit, but I didn't see any substantial rebuttals, and it's especially difficult for our opposition because the evolutionary case for these things is so easy to understand. It is so parsimonious. These things fit each other and they support the same conclusion because that conclusion is mostly accurate. And so I think that's all I have to say for my closing. Actually, I would make one point. It is my belief that creationists, especially the young Earth variety, most of them have a presupposed dogmatic conclusion that they're not even really allowed to sincerely question, let alone discard, no matter how much evidence contradicts it, nor how well they are shown that the evidence contradicts it. And that is a gaping epistemological problem in their process. And I think that the ones who don't do that, they don't tend to stay creationists long. I'm an example of that. I think there are a lot of people that are examples of that. There's a lot of examples of people who are raised in these cultures where young Earth creationism is common. They get out of that culture. They go to college. They get an education that's secular, that's comprehensive. And because they're open-minded, they realize that evolution is true. Now, that doesn't mean that they abandon creationism. And I actually don't think you have to deny evolution to be a creationist. There is different categories of creationism. Some categories of creationists deny evolution. Other categories of creationists accept it. And I think
the free time I was trying to give you, buddy.
I'll finish up with this real quick. I think the segments of creationism that don't reject evolution are superior because they don't have to reject science in order to sustain their faith. They don't have to pretend like the Bible is the scientific paper when it unequivocally is not.
All right, we're going to stop it there. We have to move on. I appreciate you guys coming to the debate tonight. We are going to get into the Q&A section now. I got some great questions for you guys, along with some funny ones here.
Mike, is my audio coming in? Because some people were saying that it was coming in a bit low on the...
Yeah, I mean, I can hear you great. I saw more comments saying I was low than anything.
Okay.
I just turned it up. If everyone wants to give one quick hello check.
Hello.
Mic check. Yeah, sounds good.
I'm
Although,
gonna.
Austin, yours was cutting in and out during that.
Yeah, yours went. Austin, you went from like real quiet to really loud.
Yeah, that had nothing to do with me. Just so everybody knows.
I just clicked a different option. Did it change at all, or is it basically still the same as before? If it's the same, that's cool.
Sounds
That
the
means
same. It's
okay. I'll
been
back to
clear the whole
original.
time.
Oh, good. Okay. Okay. Good.
Yeah, I don't think anything's wrong. Okay, so we're gonna get started here. First super chat was, excuse me, make sure this is the right set of super chats. Save today. Yep. Okay, so we got $5 from Kevin Black Well Welder. He said, for Flying Spaghetti Monster in Austin, can you name a single species that has been observed to fixate a single mutation across the entire species in one generation?
No, that's not how fixation works. It takes multiple generations.
Yeah, I don't even think it's possible for a single mutation to infiltrate an entire species in one generation. The whole part about evolution is that it takes multiple generations because if there was a really successful mutation, it's only going to be present in the offspring of that particular individual, and then it's going to take another generation for it to reach more individuals. So it's going to spread exponentially, but it's never going to take over an entire population in one generation, as far as I know.
Mike, in order to make it more interactive, are we allowed to give some comments as well, and then they can have the last word?
Yeah, if they're aimed specifically at the people, I'm always gonna let them answer first. And if you have a response, you got to make it quick because I got to get through these.
Okay. How long would you say? Because I got a timer here, like 30 seconds.
Give me like a minute at the most.
Okay,
I'll
minute. That's fine.
give you a minute at the most.
Okay, I'll start it. So it's interesting about fixation because driving evolutionary change, there's a problem called fixation equilibrium. I co-authored an article on this. It was reviewed heavily by Dr. John Sanford, very well-respected geneticist. Fixation equilibrium is this: the mutation rate, the nearly neutral mutation rate is high. Most mutations, as we've seen, especially in light of the functionality of the genome, are slightly deleterious. Well, basic population genetics says, and you can see this from Fatuma and introductory population genetics textbooks, the neutral mutation rate approximates the fixation rate. Well, the mutation rate is 100 new mutations per person per generation, which means, according to the long evolutionary timescales, that's 100 new deleterious mutations fixating every generation, which is permanent genetic damage. Since now they're fixed, selection can't remove them. That's a minute on the dot. Go ahead.
All right, thank you for that. All right, next one comes from
Can I respond to that real quick before we move on?
Yeah. I mean, this is the reason why there's a Q&A.
This is something that's been addressed over and over again. The fixation rate does not approximate the mutation rate. They're not the same thing. I think it's even been admitted to previously. So
no,
and we can move on.
Okay, well, we got that on the record now. Basil Problem says for $5 at Modern Day Debate. Last time we heard from that inadvertent pre-stream that you had been suspended from work. Are you back now? No, I actually am doing a career change, so that's why you have to send more money in the super chats when you ask dumb questions like that. All right, so next one is Kevin Black Well Welder. For Flying Spaghetti Monster in Austin for $10, he says, do you agree that humans reproduce at an average of 25 years per generation, yielding 520,000 generations since our ape ancestor? That's a 13 million divided by 25. That's what it says there.
One thing I'll say about that is that the generation length is not consistent across all times. So the length of a generation is getting progressively longer with the advent of better medicine and better technology. Modern humans don't reproduce until sometimes into their 40s, whereas if you rewind the clock, people are reproducing way earlier, or early humans were reproducing in their teenage years. So go ahead, Austin.
Yeah, I was going to say the exact same thing. A mean is not the same thing as a base rate. That's an average.
Mike,
go ahead.
1 minute.
Yeah, go ahead.
I just want to, and it's on the same issue because we're talking about generations and mutations and so on and so forth. So if I could screen share real quick and show a quote from Fatuma, I'll start my timer. So Austin Sims is just wrong. He's been corrected on this many times. Right from Douglas Fatuma, Evolution textbook. Since on average it will take 4 NE generations for such mutations to reach fixation, about the same number of neutral mutations should be fixed every generation. The rate of fixation of mutations is theoretically constant and equals the neutral mutation rate. See, what these guys don't get, including those that they look to, like Dr. Danster and Cardinal, is according to the evolutionary model, I got 30 seconds left, according to the evolutionary timescale, people did not spread out into all parts of the globe and grow to the population that we see today, 7 to 8 billion people, until recently. Most of evolutionary history existed in Africa, small isolated populations, small overall populations, which means faster rates of fixation. That's what's called fixation equilibrium. He dodged it. He did not address the challenge. That's irreparable damage every generation, which is another falsifying argument for evolution. That's a minute on the dot. Go ahead.
Neutral mutations aren't always degradation to the genome. Degradation would be more like a deleterious mutation. So neutral mutation could have no effect, or it could have some effect that's inconsequential to survival. And we see stuff like this happening with like sexual selection, where it's not necessarily better for their survival in their environment, but it's selected for because it helps them reproduce because mates find it more attractive. But a big thing to keep in mind here is also the pressure presented by the environment. So even if there's a steady amount of mutations, the amounts of change that we see over time is not going to be constant, because as the environment changes, so do the pressures which elicit these changes.
Okay.
Incredible storytelling.
There was a follow-up by Kevin for another 5, Kevin Blackwell Welder. For Flying Spaghetti Monster in Austin, assuming basic math, how do you fit 42 million fixations in 520,000 generations? This is orders of magnitudes higher than observed.
Because we don't have to assume basic math. We know that the rate is variable.
I want to respond since it's a new question technically. So notice how all we get all throughout the debate are mechanisms, but no actual modeling. That's what we've seen with the Y chromosome. They didn't even try. I got my time wrong. They didn't even try to address the heterochromatin, the palindrome sequences, the ampliconic sequences. I just slaughtered Austin by showing directly from the textbook from Fatuma that the neutral mutation rate approximates the fixation rate. I already refuted Austin and showed that the more functional the genome is, we have evidence for over 80% functionality, then these neutral mutations are actually nearly neutral. They're slightly deleterious. They're not big enough for selection to see and to remove. Okay. So the question is this: how many mutational steps are necessary to drive the differences between humans and chimpanzees in the supposed 6.5 million years from our last supposed common ancestor? And all we get is evolutionary storytelling, evolutionary philosophy. Notice how I got 15 seconds left. Notice how Austin dodged all my questions. And what he did in his closing is ask a bunch of new questions about the heat problem, which I've answered, art questions I've answered. Instead of actually addressing ours, he brings in new questions. Very dishonest way of debating. Go ahead. That's 60 seconds.
All right. Just to be fair, Matt, do you have anything to add to that?
I actually forgot what the question was.
Fair enough. We're going to move on.
Let's
Write
move
them
on.
down, Matt.
We're going to move on. I screenshot them on my phone during
I have the arguments I made in my closing listed right here.
I
They're the
answered them
5
all.
ones I made in my opening presentation.
You
So I
guys
don't
didn't
know what
answer
you're
any
talking
of my
about.
questions, and Flying Spaghetti Monster brought up new questions instead of answering the questions I brought up. Heat problem,
Well,
arc animals, we could address those. Save that for another debate.
This is a question directed
I'd
at
be happy to
Donny.
debate Noah's Ark in the future with you,
I'm
Donny.
sure you would.
All right, next one's directed
Yeah,
at
we
you,
could
Donny.
do a debate on the heat problem.
We'll hash that out another time, Austin. Not right now. At Donny by Basil Problem for $5. Donny, did you become a crazy Christian before or after you took a molecular biology or evolution class in college?
Well, I went to college for nursing, worked as a nurse for 6 years full time. And so I had good introductory knowledge on genetics and biology. And it was the genetic data that made me a confident creationist, because I saw that the driving forces of so-called evolutionary change, like mutations and selection, are limited. The majority of mutations are slightly deleterious. We have fixation equilibrium, where the nearly neutral mutation rate approximates the fixation rate. How do these guys address if you have that many mutations becoming fixed, but those mutations are slightly deleterious in light of what we know about genomic function? That's irreparable and irreversible genetic damage every single generation. They can If they want, read my article on it, attempt to refute it. I got 30 seconds here. I put out a presentation on this so-called heat problem. I got the math in there. I've written an article on it. And all you got to do is take that heat into the core, take it into the mantle, move it into space, utilize it. I got 15 seconds. Utilize it to rapidly produce coal and oil, because we got a lot of coal and oil. And so there are many heat sinks and many ways to absorb all that heat, which means the heat is actually more of a feature rather than a bug. Go ahead.
I agree that you need the heat, because you need the heat in order to get the tectonic plates moving. But that is part of the crux of the problem, because therefore you need the heat, and then you need a way to reject the heat, and it's not mathematically possible.
I've got the math in my presentation. Did you watch it, Austin, where I presented the math?
I think I did, actually.
So if the heat goes into space, wouldn't that make all the water boil? Wouldn't all the heat have to pass through the arc in order to get into space?
Well, space is a great heat sink, and you have supersonic steam jets that can take a lot of the heat into space. But the biggest mode of absorbing the heat is the core and the mantle. The core has more than enough capacity to take in the heat and then move it out slowly. Okay, and I got all the numbers in the video and in my article, so you can look at it.
You accuse us of just spinning these stories about evolution, and then you have these wild stories about the heat problem and the heat disappearing into the core. It's just
we got evidence, because we know the core. It's hard to tell exactly how hot it is right now, but it's assumed that the core has always been as hot as it is now. So there's good.
It sounds like we absolutely need to set up a debate on the heat problem.
So we're going to move on to the next question. Or actually, I want to do a shout out for who is Rich Purnell, 3328. Thanks for becoming a Final Word member. That's awesome. All right, next problem. Basil says $5 at Modern Day Debate. I'm praying that someday you'll figure out Zoom. Hashtag Ryan did nothing wrong. You know, it's not my fault that Zoom was going to jumble it all up. I was warned, but, now I have it. Anyways. Thank you for the 5, Basil. Another 5. This one's kind of for everybody, I think. But also me, because he asked me to contact the Nobel Committee. It's clear Donny has disproved evolution. DebateCon 7 in Oslo. DebateCon 7 is going to be in San Antonio, Texas in February. So as soon as tickets drop, you guys should get in on that. I think it's going to be potentially a lot bigger, more accessible situation for everybody. All right,
so next one. Was that more so directed at me?
It was. I mean, it's just like a quick jab. If you want to respond to it, go for it.
Yeah, let me just represent this challenge again under a minute. And so for those paying attention, you'll notice that all of my arguments came directly from the conventional literature. And so sure, in these papers, in the discussion section, they'll put forth untested hypotheses. The 2010 paper, the Hughes paper, they even admitted that they don't yet have a Y chromosome ancestral sequence. And so notice this, the complete sequence and comparative analysis of ape sex chromosomes. The chimpanzee and human Y chromosome is less than 30% the same. These are massive differences. And sure, they can storytell just like the evolutionists here. That doesn't mean that the evolutionary model hasn't been falsified by this data. They have yet to provide a sophisticated mathematical model showing what lineage lost what? Amplicons, palindromes, various gene families, the heterochromatic arms, the fact that the chimp Y is half the size, and where's the rate-based population genetic model? I got 2 seconds left. Let's see if Austin or Spaghetti can present that. Go ahead.
Well, one thing I'd love is that if the answers pertain more so to the question or just a reiteration of our conclusions. But I do have a question for Donny, which is, what's the real goal?
You stopped and run. You stopped. He said, based on the arguments I used today, that I should get a Nobel Prize because I falsified evolution. And I pointed out the fact that, these papers, although the authors are not rejecting evolution, if you actually learn how to critically think, which you evolutionists lack doing, then you look to the discussion, look at what their hypotheses and rescue devices are. And rather than being brainwashed and deceived into believing you're related to banana plants and that chimpanzee in the background, you can ask yourself, do these explanations work? Is there a hypothesis, right? Is there a hypothesis that's actually testable? And the answer is no. So why should we believe you when you have not even presented a sophisticated mathematical model explaining the Y chromosome dissimilarity pattern?
Yeah, Donnie, I have a quick question. Do you think that the scientific community is really going to reject evolution and embrace creationism? Do you think that's going to happen?
No. From a scriptural standpoint, we understand that people are willingly ignorant. They're going to reject the truth. Things are going to get worse as time moves on. And so, no, I'm not expecting the scientific community to accept special creation. Definitely not.
That sounds rough, man.
All right. Well,
It's not rough at all.
I just want to say I cannot control the audio fluctuations, guys. I apologize for that.
Am I being heard okay?
Yeah, it's just your mic is like quiet sometimes, and then it gets really loud out of nowhere.
All right, I would just say this real quick in response to that super chat. I think it's a valid statement because that is one of the things that led me out of young Earth creationist beliefs, realizing that evolution is a model that's used in every major, most successful college, school, university, medical research lab, hospital, agriculture business, conservation effort, even statistical computer modeling. And it seems like all that creationism can really do is put tortured nonsense on social media.
Okay, hey, Mike, because it was directed at me, to be fair, this is going to take literally 20 seconds. If you could screen share my screen real quick, since Austin Sims continues to say that he used to be a creationist. And it's funny because I answered all of his challenges in my closing and then in my rebuttal on ERVs. Let's see if he can answer one. I mean, he used to be a creationist, so this should be pretty easy.
We'll let you do this, and then the next question is going to be for Austin and Jed.
Okay, so real quick. Austin, just give me a really focused, direct answer. I've got it on screen. What is the evolutionary pathway by which an LTR evolved? And remember, LTRs contain enhancer modules, transcription factor binding clusters. ERVs have LTRs on either side. We also find hundreds of thousands of solo LTRs in the genome that can act as binding sites for initiating transcription. So anyways, the question, what is the evolutionary pathway by which an LTR evolved from a non-LTR? Go ahead.
You know dang well I can't give you the entire evolutionary pathway from abiogenesis all the way to endogenous retrovirus.
No, it's
not.
Are you
kidding me?
Just a non-LTR to an LTR. Hey, notice. Hey, to the audience, dodge, duck, dip, dive, and dodge. These guys haven't had a single answer to a single question. How is the origin of an LTR? How's that going back to abiogenesis, Austin?
You asked for the evolutionary pathway.
The exogenous retroviruses, did they come about around the same time that the first self-replicating molecule came about?
No.
Okay.
That would be part of the pathway.
Okay, so how did an LTR with all of those traits, okay, how did that evolve from a non-LTR? I'll give you another chance.
From a non-LTR? What are you talking about?
Yeah, no, we're not.
Eventually, you don't have
pose questions here in the Q&A, guys. I'm gonna cut it off there. This is for the audience to ask questions. That's why they're sending money. So this is for Flying Spaghetti Monster in Austin. Darwin said that if cells are more complicated than billiard balls, evolution is false. Why is his disproof ignored? For 5.
There are cells that are significantly more simple. There's such a thing as a protocell. There's several components that are fairly simple, like even just a membrane. That's just a lipid barrier that can be produced by just soapy chemical interactions. So yeah, no, if a modern cell is more complicated than a billiard ball, that disproves evolution. No, that's not the case.
I think maybe I'm struggling to understand the question because billiard balls might have human bacteria on them when you touch them. So there's human DNA all over billiard balls, which would make them at least as complicated as a human cell. So,
well, it's saying if cells are more complicated than billiard balls, evolution is false. That's what. Yeah, well, that's what he's saying. Darwin said.
If we ignore all the biological debris that might get trapped in a billiard ball, that's not really surprising because billiard balls are not alive. They're inorganic material. So, of course, organic material is going to be more complex than inorganic because it's carrying out functions.
Mike, I'll take 30 seconds, not a minute this time. Everything about the cell, the sophisticated nature of it, there's what's called mutually interdependent systems. These guys can have the last word to dodge again, okay? Because I noticed Spaghetti said that he's struggling to understand the question. that should be on the thumbnail. It's a picture of him with that quote, because that's been him for the last 2.5 hours. So there's something called mutually interdependent systems. Okay, Spaghetti, let's see if you can answer this one. What came first, DNA or DNA repair enzymes? Because it turns out, incredibly, that we actually could experience up to 1 million DNA breaks in a single cell in a single day. And luckily we have DNA repair enzymes, okay, incredibly designed DNA repair enzymes. The information for those are actually encoded in the DNA. So if you say DNA repair enzymes came first, well, wait a minute. You need the DNA to encode the information for the DNA repair enzymes. If you say the DNA came first, well, wait a minute. Life would go extinct because you need DNA repair enzymes to fix the DNA break. So which came first, Spaghetti?
I would guess DNA, right? Because the repair enzymes would need the DNA in order to exist. But of course, everything evolves codependently. So it's like, oh, which came first, the chicken or the egg? But of course, they both evolve. Well, other animals lay eggs, of course, but different things can evolve, at the same time. So it's mutual evolution.
What's the best paper you can cite so I can look into that story?
I don't have any papers in front of me.
Ask what you're talking about, because you're just talking about irreducible complexity.
Is that irreducible complexity, mutually interdependent systems? He said DNA, but wait a minute. DNA is not going to last even a few generations if you don't have DNA repair enzymes to fix all the DNA breaks. And there's no such thing as a proto cell or a primitive cell. That's all storytelling. That's all Spaghetti has.
Super chats to get to. I'm sure we'll find a better question to hang on for a little longer. This is not it. $2 from an honest doubter. How about, quote, more closely related than any other? And I think this was in reference to the hyoid bone discontinuity questions. Doesn't require a response if there wasn't really a question there.
You can move on.
$10 from Basil Problem again. He said modern day debate. Something happened in room 627. That's number one, premise one. Premise two, Andrew Wilson hugged many DebateCon participants. Therefore, Andrew Wilson hugged at modern day debate in room 627 by modus ponens. That is awfully deductive, Basil. We appreciate you, your super chats. 627 was just an extra room that was refunded, if anyone was wondering. I don't know how it became a meme, but it was just like booked on accident that we didn't end up using, is from my understanding. And we'll go to the next super chat with that. This is from Kevin again for Flying Spaghetti Monster in Austin. If humans came from great apes, can you explain why the same mechanisms can't be used to derive where male or female came from, or came first?
Well, we've already mentioned how humans are great apes. So saying that they came from great apes, I mean, that is technically true. Male and female reproduction or sexual reproduction, it's not like one came first, the male or the female, right? So you have asexual reproduction was the primary driving force of reproduction at first, and then sexual reproduction evolved from that because it's very beneficial to immune systems because you're sharing your DNA with somebody else. But that didn't make asexual reproduction immediately go away. And there are organisms which reproduce sexually and asexually even now.
So a minute on that one. That's interesting.
Before you go, Donny,
I can't let you respond to all of them. To be fair, was just directed.
Okay, how about 20 seconds? Because he said we are great apes.
It is directed at Jed and Austin, so I'm gonna have to let Austin go first there, and then I'll give you 30 seconds.
Yeah, I find the question a little confusing. I'm leaning towards saying female because every human starts off female and then diversifies in fetal development. I would hope most of the audience would know that. But yeah, no, the question is not exactly clear to me.
Yeah, he was a little bit vague. Go ahead.
30 seconds. So he asserts we're great apes. Okay, taxonomically, the great apes did not classify themselves as great apes. When we actually look at the differentiating data, I know you're excited, Austin, calm down. I'm going to further destroy your model. Okay. So here's the thing. When we look to discontinuities and genetic level differences like the Y chromosome, like the hyoid, like these major gaps, that actually suggests that no, we're not great apes, even though they can continue asserting that we are. Go ahead. That's 30 seconds.
All right. There was a follow-up from Kevin for you guys that said,
For FSA and Austin, Genghis Khan is an ancestor for 5% of humanity. His genes have not fixated the human genome in 400 years. What is the explanation?
You're expecting an entire human's genome to fixate? Every one of his offspring would be a clone of. Your questions are not making sense. I'm sorry. They're very confusing.
Yeah, Kevin, be more clear. Send more money.
It almost seems like he's trying to say, if this one person is a common ancestor of a lot of people, then he should be a common ancestor of everybody, and that just doesn't make any sense.
Okay, well that's a pretty fair response. This one's at Matt, finally. What fraction, Basil Problem says for $5, what fraction of the Nobel Prize do you think you should share, Matt?
Oh man, I'll take any. I guess it would make a little good stand on my counter, so when people ask where it is, I can just show it.
Donny, are you okay with that?
We can rent it. We can borrow it. We'll just trade
Well,
it.
You get it on Tuesdays.
If we get the golden Nobel Prize, can we give Austin Sims and Flying Spaghetti Auditor? Can we give them a golden dodgeball for all the dodging they did tonight? Would that be fair?
I would love it.
We'll have to get another.
I would cherish it.
Confirming
Perfect.
whether or not we can or not.
Yeah, we'll look into it.
So Kevin also said to you guys over on the left here, FSA and Austin, you claimed a difference is not a discontinuity. Can you provide the exact difference between the two? Exact.
Yeah, a discontinuity would be a level of difference that cannot be explained by common ancestry. Like if humans and chimps had the total percentage of telomere to telomere nucleotide identity sequences that they shared was like 5%, that would be a discontinuity. But it's not. It's well over 86% in the entire genome, 98 to 99% in the protein coding base pairs. That's not an unbridgeable gap. All of the gaps Donny brought up are very bridgeable.
My turn.
I'll also add that if humans and chimpanzees or humans and any other animal are not super genetically similar, let's say that humans and some bacteria are only 5% similar, that doesn't mean that we don't share a common ancestor, because every living thing on the planet shares a common ancestor. And the more dissimilar we are between ourselves and some other organism, the further back in time you have to go before you reach said common ancestor.
Yeah, it's very
Mike,
simple.
can I screen share real quick for my 30 seconds?
Very quick response. Go ahead.
I'm always going to respond, just warning. This is a debate. I'm not letting them get away with assertions and storytelling. So yes, these are major discontinuities. They could dodge, duck, dip, dive, and dodge all they want, okay? Anybody who's actually objective can see here on this. They just don't get it, do they? Just right over there. It's the differences that make all the difference, okay?
Why are you ignoring all the similarities in the X chromosome? Look how similar those X chromosomes are. They're almost
You're going to
identical to each other.
respond to every question, then we're going to have to respond to your responses. This Q&A will never end.
Because
That's not going to work.
Response each. You can even
That's
have
not
the
gonna
last
work.
word if the question's for you, okay? I got to take 15 seconds back because these guys were excited. Control yourselves, guys. Okay, so notice this. The Y chromosome between humans and chimpanzees, again, all they've offered is mechanisms, no model. The chimpanzee Y is half the size. It's missing palindromes, ampliconic sequences, the entirety of its heterochromatic arm. Austin just conceded the debate because he doesn't want to admit it, but this here is a major discontinuity that common descent can't account for.
It absolutely is not.
Okay.
That's your assertion. Demonstrate it.
It's also
You
interesting
think you
how
can't
we're
account
spending
for
so
a
much time-
2% difference? Of course you can. Ridiculous.
We're spending all of our time fixating on the Y chromosome, but not on the X chromosome, which
Exactly.
is way more similar. So why doesn't that one matter at all?
How come you're
Because
always
the
taking a
similarities
nap?
don't
How
matter
come
and
you're
the
always
differences
taking a
are what
nap? when
cherry
I'm talking?
picking.
matter.
It's the differences that make all the difference. You guys can't
Right.
address the discontinuity, so you keep going back. You keep running back to the similarities.
Yeah, you're saying look at the 1% that's different, and we're saying look at the 99% that's the same.
That 1% is the entirety of the Y chromosome, which is spaghetti. How big
So
is the Y
it's
chromosome?
still 1%.
No, listen,
Still
I got a
tiny
basic
difference.
question. Austin, how big is the
Tiny
Y
difference.
chromosome? How big is the Y chromosome?
Microscopic.
40,000
that note-
genetic base pairs.
40,000? No wonder why you guys are dodging.
All
No,
right,
Mike,
guys.
Mike, this is important.
I get it, but I have to moderate and move this on. I
No,
appreciate
it's
it.
not just 40,000. It's in the millions, 65
All right.
million.
It's
That's not a small
significantly
number.
smaller than the other
I'm gonna have to mute.
chromosomes.
If we can't go on, guys, don't.
I
make
can't
me mad.
memorize every number.
No, I'm just correcting. It's not just small or minor. It's huge. Go ahead. I'm sorry.
It's smaller than all the other genome,
Alrighty,
all the other chromosomes.
Now this is relevant. Basil Problem says for $5 at Modern Day Debate, Raw Matt is not a woman. He deserves to be heard. Matt, would you like to respond to that? You don't have to. And Donny, you don't have to either. just
I love
saying.
my wife, and I'm happy for her to say anything, and I'll hear it.
Okay, well, that's fair.
Well, that's good.
All right, now for $49.99, Big J Row 5. I believe I met this gentleman at DebateCon. Very cool. He says, "Stop looking at my cytochrome, bro."
Your cytochrome C is showing.
Yeah, you all chuckled, so I owe him $25. Damn it. Okay. Basil Problem again for $5 says, to the debaters, humbly, is it possible you are low information on this topic and your opinion should be met with skepticism? That was everybody.
Okay, I'll answer first. No, because everything we said can be backed up with the conventional literature. We've probably screen shared papers and questions and data points directly from the conventional literature for 85% of the debate. And the reason why Spaghetti and Austin, not to be rude or anything, the reason why they're dodging the Y chromosome is because we just heard from them that they thought the Y chromosome was only tens of thousands of base pairs or sequences. I mean, the Y chromosome is 65 million, and yet when you compare it with the human Y chromosome, it's less than 30% the same. All they have is storytelling, evolutionary philosophy. I mean, they could try again in the future, but they did not meet their burden of proof here tonight, and Mike knows it. I can see it in his face. Go ahead, Mike.
Yeah,
unlike
unlike Donny, I'm happy to show a little bit of humility and fully admit I'm not an expert on biology. I'm not a biologist. I don't have any degrees in the subject, nor do I think anyone on our panel has degrees except for Austin, who's in the process of getting a biology degree. If I might be mistaken about that, but feel free to correct me. I'm just a layman who feels interested about the subject, and feel free to take everything I say with a grain of salt and go look up answers for yourself. Don't take anything I say as gospel.
Well, you didn't provide them any answers, so yeah, of course they're gonna have to go look up answers for themselves. You certainly didn't provide.
Donny, contain yourself, okay?
Yeah, I know you're excited, Donny, but please.
Yeah,
Hey, listen, you gotta pick your own liner.
I was
Calm it
laughing
down.
at the chat, just to be clear, a second ago. That was what you saw on my face. There's some
So
funny
my answer to
people
this
there.
question is to repeat the scientific epistemology. Evolution is a quantifiable, falsifiable, possible solution to unanswered questions about unsolved problems in our attempts to study and better understand natural history in the universe using what we learn to improve human existence. Is it possible that parts of that model could be better formulated? Probably. Is it possible that evolution doesn't occur? Absolutely not. Species immutability versus mutability has been out the window forever, and even Donny admits that.
All right. Matt,
All right.
you got anything to say? Go ahead.
Yeah, because Austin Sims said earlier that there's no functional purpose for young Earth creation. It's not used anywhere. Actually, it's used in the NIH, the National Institute of Health, the largest health organization on Earth. They use Mendel's accountant, which is actually John Sanford, young Earth creationist model for how mutations travel through a population. And the FBI actually also use this mutation rate as well to track and convict killers and go backwards in time. He solved the Romanov murders. And here's the biologist from the PLOS biology himself. He said, in fact, over the last 100 years, almost all of biology has proceeded independent of evolution. Molecular biology, biochemistry, all of it doesn't take evolution into account at all. So I just wanted to throw that out there because, we gave you the observable, testable, repeatable evidence of mutations that explain the diversity of life on Earth. And if all the experts are wrong, then so be it. But they're actually using these things in the field. So I guess I could be wrong, but so are they then.
you're talking about one part of one paper, which I'll admit to you, it has some validity, but compare that to something like Watson and Crick's paper that's been referenced 20,000 times. It was a high impact paper. It's influenced high impact papers. That is the majority, the overwhelming, vast majority of the corpus of scientific literature on this subject. I think creationists have contributed maybe 16 papers to the corpus of literature, and none of them have significantly substantiated creationism or falsified major aspects of evolution.
That's because we're not actually allowed to actually pass peer review or even really get in. And I know because I tried, and this was their rejection letter. We consider that the work does not represent the type of advancement that PLOS biology seeks to publish. So they wouldn't even look at what I sent them that was creationist oriented. So of course you're not going to find the information.
I think it's more likely that they just detected bad science.
Or I could name several young Earth creationists who have published in the secular journals. Dr. Rob Carter comes to mind. He published a really good paper, an excellent paper on mitochondrial DNA, and you can look at the diversity and you can see how it correlates with the biblical Eve just a few thousand years ago. Now, the thing is, though, in that paper, you'd have to read between the lines. You'd have to critically think through it, because if he were to just come right out and say, and by the way, this refutes human evolution and confirms young Earth creation, well, of course, it's not going to get published. But you could publish it in a way, as many young Earth creationists have, that shows you the data, and anybody with an objective thinking mind can look at that data and think, wow, that supports separate ancestry.
All right. For another $50, JRO5 says, so my cane corso is related to an even more badass wolf? Based.
Yep. That's right. All living things are related. Absolutely. My cat is my cousin. I have 3 cats, and we're all cousins hanging out.
Why do
it's
I
great.
feel that that's going to get clipped?
Are you related to a banana plant?
I am absolutely related to a banana plant. I am the cousin of the bananas I eat, and I'm proud of it.
All right.
Are you related to a mosquito?
I am related to a mosquito. If you can think of any living thing, I'm related to it.
Grass?
We're related. Rats, mosquitoes,
grass.
bacteria, mushrooms, rats, mice, elephants, pineapples, everything. Every single living thing on the planet shares a common ancestor. We're all related. And that's the beauty of evolution.
Yeah, the beauty of evolutionary philosophy, since all the evidence we presented tonight actually contradicts your belief that you're related to grass.
Right. Pseudonym 8344 for $2 says, SFT, let's go. Interesting, though. Maybe thought. Matt, can you blush?
Blush. I don't know.
I'm the one
maybe.
who blushes. Matt's always calm, cool, collected. It's the good cop, bad cop routine, right?
Maybe if somebody goes over there and tickles him.
Now, this next super chat has nothing to do with the debate, so take it or leave it. He did give $10. JRO5 says, should we outlaw marrying first cousins in the United States?
Oh my God.
Isn't it outlawed already?
Can we not respond to that one? Let's just skip that one.
just doing it.
Is that the last one?
Well, I will say Spaghetti said he's related to mosquitoes and grass, so that's his cousin. Should we outlaw a marriage between spaghetti and grass and spaghetti and mosquitoes? Or where do we draw that line? What about a chimpanzee, for example? That's a pretty close cousin, right?
I think interspecies marriage is already illegal.
Is it? Okay, well, that's good.
So yeah, I just looked up whether first cousin marriage is legal in the United States. In 18 states, it's completely legal with no restrictions. In 8 states, it has some restrictions, and in 24 states, it's completely illegal. So
I bet you we could guess the states that are.
It's funny because the human evolution model would say that we all spread out of Africa, seized dominion over the planet, and invented the space shuttle through inbreeding. Look up the out of Africa theory. You have thousands of years of the human population existing in isolated tribes, small population, severe inbreeding. But we're supposed to believe that highly inbred, genetically degenerate population of roughly 10,000 in Africa a couple 100,000 years ago suddenly spreads out in all parts of the planet. They seize dominion over the globe, build these laptops. We're having this nice, respectful debate over the space shuttle. Now that is a wild story, gentlemen.
You realize that creationism
I don't-
poses the exact same problem, but times a million, because you reduce all humanity back to two individuals. So think of the inbreeding with two people. You got people marrying their sister and brother, Cain and Abel, or
That's before the
Seth or whatever, marrying his sister who he lived in a different town, and then all of Noah's kids had to, Noah's grandkids had to reproduce with their cousins. It's a big mess. There's way more inbreeding in the biblical model than there is in the evolutionary model.
Well, you must have been taking a nap, Spaghetti, when I explained this. You're the one who has the problem because you explain the majority of DNA variations being the result of mutations. We, as evidenced by genome-wide functionality, explain the majority of nuclear autosomal DNA variation as being the result of created heterozygosity. So if you don't have mutations to come to the forefront, like in your model, to lead to rapid genetic degeneration and disease, then no, we don't have a problem. You have the problem.
You're saying
Noah's drawing board.
You're saying Noah's three kids had these three properties, if I'm following right. And so they got all of those properties from Noah, right? So which one did Noah have? Did he have all three?
Well, Noah would have passed on his Y chromosome to Shem, Ham, and Japheth. There might have been a couple of mutations there, or in biparentally inherited DNA, he would have passed on some mutations. But from creation to the flood is only a couple hundred or a couple thousand years, and so you wouldn't have as many mutations or a high genetic load in Noah's family like we see today. Plus, it would only be an issue if it were a multi-generational bottleneck. Your out of Africa, your false out of Africa model is a multi-generational bottleneck. It goes on for thousands of years, which means that's thousands of years of deleterious mutations that have accumulated for thousands of years to come to the forefront, leading to disease and accelerated genetic degeneration. But the flood bottleneck is one generation followed up by rapid and exponential population growth. Look it up. It's not a problem.
Okay, we have 6 super
Oh,
chats
it absolutely
left,
is.
Ooh, that's bad for you.
we got 6 super chats left. I don't know how much time everybody has. We can get through these pretty quick. If anyone has any last ones that
I'm
they'd
here
like to
till
get
the
in,
end.
if anyone has any last ones they'd like to get in, it's now or never. Okay, we got one more just from me saying that. Nice. All right. Any more than that, then we'll keep it going. But otherwise, let me ask this next one, which is at Donny from Basil. Why were you a nurse and not a doctor? Isn't nursing for women?
Hey, what does this super chatter have against women?
Dude, I think he just likes spending money. And he's trying to get to
Male
the
nurse
bottom of everything.
Male nurse does remind me of Meet the Parents. When Ben Stiller, they thought he was the doctor. Thank you, doctor.
Yeah.
Oh, no, I'm just the nurse.
I'm
Lord
actually,
Flocker.
I heard someone say it in the comments, but I'm actually getting quite fond of Basil. He asks some pertinent questions sometimes. Not that one, but some other ones. Yeah.
Did you say you're quietly fondling Basil?
I did not, but I could understand how you could hear that because you're listening for it. So just let's try it again with the next one. I'll be clearer. JRO5 says 99 for $9.99. It is outlawed relating to the cousin marriage. How do we convince all the, I believe this is supposed to say Muslims, but YouTube might not have let him comment that, to stop making their first cousins besides just arresting them?
They have their own law. They have Sharia law. They don't care about ours. They got their own rules going.
I'm not touching that with a 10-foot pole.
That's fair. Donnie?
I'm good. I'm getting tired. So
all right, we're almost done. Kevin
One thing I'll say is that our world would be way better without Islam.
Okay, that's probably what he was trying to get. Response-wise. For Austin, this is from Kevin Blackwell Welder. Just to clarify, you think math and statistic don't apply to evolution? Do you think statistical averages don't apply, or they do?
I think there's a fallacy in representing a mean as if it's a rate. The rate is variable. The mean is not. Pretty simple.
Okay. Next one. 85 that's spelled AIDYPHIVE 45, says, where is creationist sophisticated mathematical model for evolution?
Are you saying us creationists need to provide a mathematical model for evolution, for common descent?
That's what it sounds like. Yeah. Where is
that's
your
confusing.
mathematical model for evolution from the creationist perspective? If I'm going to.
Don Sanford has done one of those. I can show you right now on the created heterozygosity model, if I can find it, where he did actually run a comparison between the two. Just give me a minute from my thousands of slides. And Donny,
you go ahead and take that minute. Maybe Donny can answer this while you do. What is created heterozygosity? I've heard this term used like 4 or 5 times now. Can one of you explain what that means to me?
Yeah, no problem. Created heterozygosity is the model that suggests Adam, Eve, and the original created kinds would have been created in a state of DNA diversity. So they would have had front-loaded DNA differences within themselves and then across. So in the animal kinds, they'd have pre-existing DNA differences. And that makes sense theologically, because when God said, be fruitful and multiply, it wouldn't make any sense for him to want that to be carried out through cloning. So Adam and Eve being created with pre-existing, so heterozygosity would be a higher state of DNA diversity, right, Austin? Yeah. Homozygosity would be a lesser state of DNA diversity. So in a nutshell, it's that Adam and Eve were created in a state of heterozygosity and not homozygosity.
Okay, so is it accurate to compare this to like the young earth creationist argument that Adam is created fully grown and maybe the universe is created with light from distant galaxies already in transit towards us, such that it fits the timeline of conventional science?
No, because the timeline we get, and I wrote an article, as I said earlier, on fixation equilibrium, but also on the neutral mutation rate approximating the fixation rate as it pertains to from today to the flood. Okay. And I got the numbers, the math in there to answer the questioner's comment. But with created heterozygosity, that doesn't have to do with uniparentally inherited DNA, like Y chromosome mitochondrial DNA, Austin, which lines up with the biblical timeline. Biparentally inherited DNA, where we actually have these DNA differences, we're applying it there, but it's not ad hoc. It makes testable predictions on DNA function. If created heterozygosity is true, we'd predict that the vast majority of the genome is functional. If it ain't true, like the evolutionary model would suggest, the vast majority of the genome would be non-functional. Does that make sense? Mostly junk, 10%. I think I've heard Dan Stern Cardinal say up to 13%. So that's a head-to-head prediction right there that we could test.
Donny, when you say that Adam was made
it's just not a common term. I haven't heard it a lot.
When you say that Adam was made with,
I haven't heard it that combination of terms. Sorry, go ahead, Jen.
Yeah, when you say that Adam was created with more diversity, what's the mechanism for carrying that diversity? Are you suggesting he had like more than a normal number of chromosomes, or where does the diversity come from? And how is it passed on to his children?
It's not an abnormal amount of DNA diversity. Like even today, we have DNA differences within us, a few million. Okay. So the math has actually been done. You can look at common variants. So the question that evolutionists sometimes ask is, well, how do you know what is a created allele and what's an allele due to mutation? Well, a created allele would be a common allele where it's high in frequency, where basically everybody has it, non-disease causing, beneficial in some way. And if I remember correctly, I have a presentation on it, but at the top of my head, I'm pretty sure there's about 10 million common alleles on the planet. So at the flood, if there's a bottleneck, okay, Adam could have had 20 million pre-existing DNA variants. At the flood, you lose half. And then that 10 million spreads out to all parts of the globe at the Tower of Babel. And so you can track back, you can do the math and see how many DNA variants could have been in Adam. And it's not an overabundance, as in he'd be like genetic overload or something like that.
Right.
But the number of genetic base pairs would be larger.
Well, it just goes back to basic alleles, right? If you're capital A, lowercase a, capital B, lowercase b, what are you? Genetically heterozygous. If you're capital A, capital A, capital B, you're genetically homozygous. There's no DNA diversity there. So take that concept. capital A, lowercase a, capital B, lowercase b. Apply that to millions of positions in Adam's genome, and you have Adam existing in a state of heterozygosity rather than homozygosity. And then from there, if you apply that to the created kinds, explaining all the species is easy, because then you get rapid speciation, since what happens every generation, you get recombination, where all of those pre-existing DNA variants are shuffled up to produce novel chromosomal combinations.
All right, well, we're getting more super chats as we talk, so it looks like I will keep reading them. But I do want to move on real quick. Matt, did you have that model?
Yeah, yes, I found it real quick.
All right, well,
I have 4 pictures to show.
I'll let you screen share real quick, and then we'll move on to the next one.
Okay, this is John Sanford, and he is a geneticist who invented the gene gun, published 70 papers. He wrote a paper specifically called the Adam and Eve Design Diversity Allele Frequency. When he tested the Y chromosome, he shows using over 1,000 different genomes that the extreme rarity of high-frequency alleles in the Y chromosome, showing that it's young. And he compared all of these minor allele frequencies to the evolutionary model. They did not match up. So this would be the answering your question directly. Has it been tested with the evolutionary model? And it has, and that is how you can find that study.
Awesome. Thank you for that. Okay, next one says JRO5 for 20 says, sounds like somebody didn't get a hug from Andrew in room 627. Okay. I don't know.
I didn't get a hug. I'm disappointed.
That was supposed to be referring to you then specifically.
Yep.
And then he clarified here for another $10. JRO5 says, no, sorry, you did not meet me at DebateCon. I run JRO's comms once contract is made. So I was mistaken, everybody. I apologize.
I'm assuming these are DebateCon inside jokes.
As far as I know, that was a joke made here in the stream, because I said I think I met him at DebateCon. So he was like, no, you didn't meet me. I run this person's communications once the contract is made. That's about as much as I have as well, Donny. The next one is from Basil again for $2, saying Donny is comically sure of himself. Would you like to address these allegations?
I do like comics. I got a whole comic collection over there.
All
Superman,
right.
Batman.
I actually think I can add something here. If I can ask Donny and Matt a question that might help demonstrate that they're not comically overconfident. Donny, Matt, let me pose a hypothetical to you. Say some point in the future, you're not a young Earth creationist anymore. Tell us the story of how that happened.
Sure. In a debate, you guys actually address the challenges, the questions, provide me a sophisticated mathematical model of how the Y chromosome dissimilarity differences could accumulate the heterochromatin, the palindromic sequences, the amplicon differences. Show me why the majority of the genome is junk. Because like I said in my opening statement, when evolutionists ask me, what would it take for you to believe in evolution? Well, show me, since here's the thing, genes, traits, and genetics, right, Austin? That's what's inherited sperm and egg. That's the most direct way to answer the question of ancestry. Fossils are not inherited sperm and egg. Geography is not inherited sperm and egg. So when it comes to a head-to-head prediction, creation versus evolution, we predict that the vast majority of the genome is functional. You guys would say that the vast majority of the genome is non-functional, about 10% functional. So show me that, and then I would be forced to admit that, okay, the genomes of living organisms reflect a long history of common descent. Does that help, Austin?
So what about knockout experiments where portions of the genome are removed and it doesn't have an impact on how the phenotype is expressed?
That's a good
That's
question.
like having backup tires in your vehicle. Those backup mechanisms showing that they can take over from knocking out different parts of the genome, and it has a redundancy built in after redundancy. That screams design to me. And what would get me, I guess, if aliens came and said, "We created you, you idiots. Here's how it was done." I'm done. I'm done. That's the end of young Earth creation, probably.
Yeah, like the movie Prometheus. If that turned out to be true, we had good evidence for that. But to your question, Austin, it's a good question. Unfortunately, many non-coding RNA genes they're only expressed, some ERVs as well, they're only expressed under very certain conditions. For example, various developmental windows, you have genes like ERVs as well, DNA units, that are expressed and functional. Then when they're done with their job, they're turned off. So hypothetically, you could actually snip that out and there'll be no effect on phenotype, but it's not because it's not functional. It's just because now it's not needed. It's not expressed. So that's tough. You basically need unlimited sensitivity to determine that the majority of the genome is junk through knockout experiments. And for ethical reasons, thank God, we haven't performed knockout tests on humans. So that'll be a difficult one to determine.
Okay, we're going to move on to the next chat so we can maybe get done by 11:30. So to recap, J-Ro had said earlier, cousin marrying is outlawed. How do we convince all the Muslims to stop making their first cousin, blah, 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 blah. So just to recap, because this is connected to that one. For another $10, he says, Andrew Wilson just argued that marrying first cousins for 1,400 years creates populations that are 70 to 85 IQ.
I think inbreeding is generally bad for genetic diversity, especially consecutive generations of it. So that's just a biological fact.
Mm-hmm. I've only read one article that was for it. It was called "Go Ahead and Kiss Your Cousin," and I read it years ago. And it was for
Who wrote
cousin.
that article? Who wrote that?
I don't know. I'm gonna look it up though.
Put it in the chat.
Oh my god.
We'll have some people looking at that. But so
Didn't Charles Darwin marry his first cousin?
He did, and interestingly, one of the only entries he ever put into one of his scientific journals that wasn't about science was a pros and cons list where he was debating whether or not he should get married, and the fact that he was in love with and planning to marry his first cousin was one of the things that showed up on the list. So that's part of the reason we know that did happen.
Well, this
It's an
one,
interesting anecdote about
another
Darwin.
follow-up from J-Ro, and this one is specifically towards Donny. For $10, says, can standing for truth give the definition of a woman? Okay, Sergius 2186 for $5 says, just one look at the flagellum motor. How can one assume this is the result of randomness? Open your eyes, man. Someone engineered all this.
The flagellar motor is the poster boy of irreducible complexity, and it was completely smashed, annihilated at Kitzmiller versus Dover. The guy who came up with irreducible complexity, Michael Behe, and who used the bacterial flagellar motor as his example, he had to admit on the stand that he hadn't read over 50, 60 something papers about bacteria that had flagella that had less than the total 50 parts he says you need to have a working flagella. There's bacteria flagella that have even just a tenth of those parts and are still able to use what they have there, even as what a motor flagella is used for locomotion. They just do it by acting like a jet pump. There's an even simpler version where it's just a protrusion, basically, and it still has a use in that it can exchange things from the cellular environment with the internal cytoplasm. So it's just wrong. I don't think anybody has ever demonstrated something that's actually irreducibly complex. It doesn't exist.
Okay.
But Austin, what about the human eye?
All right, that's facetious. We're going to move on. So 85 for $5. So no sophisticated mathematical model from the creationists then. Matt?
Yeah, they just got to rewind. That the math is in that model. He literally wrote Mendel's Accountant, which is a mathematical simulation and model based on math, and it's used by the NIH. You can go directly to the NIH website and type in Mendel's Accountant, and you can run that as well. Do population genetics with it. It follows cancer through the population. That's how the institute uses it. But what was it called again? The direct paper title is Adam and Eve Design Diversity and Allele Frequencies by Dr. John Sanford and Robert Carter.
And on top of that, Dr. John Sanford, you can look it up on my channel. It's gotten a lot of feedback over the years. He actually presented, Young Earth Creationists, he's a geneticist. He presented at the NIH, National Institute for Health, clearly not a creationist organization. And he presented on the problem of human genetic deterioration. And he went over all the possible rescue mechanisms for it, synergistic epistasis, super beneficial mutation accumulation, so on and so forth. And he demonstrated in a non-Young Earth creationist institution that problem hasn't been solved.
Okay, we have one super chat left, you guys.
Yeah, let's go.
We made it.
Yeah,
we made
to
it.
the end.
With a mini political debate in the middle of it all.
Oh yeah, got a little sociological at the end there too. So JRO 5 again, base $19. He says, or $20. He says everyone should judge the scientific aptitude and rigor for the truth based on their answers to my woke outing questions. Then sunglasses emoji.
Was there a question in there?
It was a super
No,
chat.
it's a very loaded statement.
It's basically just saying to pay attention to how everybody responded to the last super chat.
I would like to make a response to that.
Go for it.
If you're going to claim that, then what makes me claiming creationists like Todd Wood or Kurt Wise or Katrina Cook, creationists who admit that there is evidence to support evolution, that the body of evidence we have is sufficient and is confirming and matching evolution, but they have chosen to reject evolution based on a faith choice. Kurt Wise, Todd Wood, Katrina Cook, all of them have admitted that. And I actually think that's a more convincing model of creationism because it's honest.
But then how come you
It's
don't
honest
accept
in its epistemology.
But then how come you don't accept Todd Wood, who is, in my opinion, perhaps a little too generous to the evolution position? It's interesting. Even someone like him who wouldn't attack evolution aggressively like me, he has done a lot of work in what's called statistical baraminological analyses. And you can watch his talks where he says using statistical analyses, he can actually determine what is and what isn't related. And based on these discontinuities and gaps in his analyses, he has said that he can confidently say or claim that the Australopithecines cluster in their own group, your great apes cluster in their own group, and then extant humans alongside extinct humans like Neanderthals, Heidelbergensis, so on and so forth, cluster in their own with all these gaps and discontinuities. So even him being very generous to the evolutionary position, he goes, runs these analyses, and he discovers, wow, separate ancestry.
I'm familiar with baraminology to the extent that it's available to the public, and I'm watching it closely. I want to see more of it emerge before I comment on it.
Okay, that's fair. Well, and I'll say this too for the RJ Downers of the world. They've done statistical baraminology, Matt, as you know, on the mammal-like reptiles, the synapsids or therapsids. And amazingly, even in that category of so-called transitional like forms, very interesting creatures, right? Blending the features of reptiles and mammals, they've also discovered discontinuities and gaps, separate ancestry.
Okay, well, I will let Jed say something if he wants, and then we're going to get into just giving everybody your handles and everything. Thanking you guys.
Yeah,
Great debate. Thank you, guys. Thank you, Donny. Thank you, Matt.
Go ahead.
I had one final question for Matt and Donny, which was, if you lost your belief in God, would you still reject evolution?
If I lost my belief in God, would I? Well, it's a major hypothetical. You know, if aliens came down and took me to their planet called Krypton, and it turned out I'm a Kryptonian, I mean, it'd be interesting.
I know it's a hypothetical, but
probably I'd still reject evolution because, you know, there's a lot of non-Christians that also reject evolution. And then they look at things like ancient alien theory, or, you know, there's a lot of positions outside of Christianity that recognize the problems with evolutionary theory.
Donny, the Kryptonians can't come and pick you up and take you back to Krypton. It got blown up, okay?
That's a
Get
good
your
point.
facts
See,
straight.
that's why
Get
it's a
wrecked,
hypothetical.
nerd.
That's why it's a hypothetical.
I'm just messing with you.
Maybe we could do some multiversal story where Krypton, managed to survive, and now they need me to fight Brainiac or something. Dark side.
Well, thanks for being a good sport.
That's where Austin and myself would get along. Dragon Ball Z, DC Comics, Marvel. We just don't agree on origin. Exactly. You should see.
I'm gesturing at my Vegeta right now.
You love my Vegeta.
Whoa. Keep it TOS, brother.
So it's fiery, but I enjoy the passion, and I like debating these guys because clearly they got thick skin. We can come in, we can have a good sparring match, and shake hands at the end of the day and say, "Hey, that was fun." And I thought it was a lot of fun.
It was.
I enjoyed it.
All right, we'll start from the left here, which is Standing for Truth. Tell everybody how to find you. Tell them about your book, and then move on to the next one. Would be Matt.
You sound like my mom when we're out at family day. Donny, tell them about your book. Tell them about your book.
Come downstairs.
Come downstairs. Mom, not right now. Yeah, no, you can find us over on Standing for Truth, standingfortruthministries.com. Endless material, endless content, articles, books, videos, interviews, debates. I've also moderated, over 400 debates on all sorts of topics. I've been debating here on Modern Day Debate for the last 5 years. I think I've been here probably about 30 times. I always enjoy James Kuhn's channel here. So best ways to find us, Standing for Truth on YouTube, standingfortruthministries.com if you want kind of one place to find everything. So Matt, anything you want to add, brother?
Sure. I have the channel called Young Earth Creation. It's mostly just a backup for when Donnie gets his taken down one day. And I have a website called Creationist Clothing, and we're about promoting knowledge. So all of our information and our books are free. You can go to the articles section, and the very first link that you see at the top is all of our books. You can just click on, and it'll take you straight to the Google document, so you can learn about our model, how we view the flood. Because Donnie and I have our own models. We have our own views of different things. We don't even agree with all of the different creationists out there. We follow, I would say that we follow evidence more. That's very bold, but I'm going to say that. I'm just going to go out there. So that's
we like to engage the critics. So we're going to have different approaches because a lot of creationists that are not engaging the critics, I always encourage that they should be. Debates, discussions, response videos. So we're going to have a different approach to engaging certain arguments like ERVs. So for those that want to see more detailed answers on this, they can see my book. Austin, I'm happy to have a more informal, laid back, relaxed discussion at some point on ERVs. If this was the formal, more heated, fiery debate, I saw you in the live chat before the debate, Austin. And so I thought, okay, let's fight fire with fire in fun, right? But if you want to have more of a civil discussion, just more of kind of an informal, laid back discussion on ERVs, happy to do it in the future.
All right, that being said, we're going to let Jed go ahead and close up and tell us about himself. If you got a book, YouTube channel, anything you want to promote, go ahead, now's the time.
Sure. Well, I've never written a book, but I do have a YouTube channel, Flying Spaghetti Auditor. Same handle on Instagram, Flying Spaghetti Audits on TikTok. I do mostly shorts, so I make comedy shorts about religion, primarily Christianity, a little bit of Islam. Basically just poking fun at the contradictions, pointing them out, exacerbating the moral atrocities within the Bible, pointing out how the Bible is incredibly contradictory. It's a source of moral bastardization and pseudoscience. So I really just like ragging on the Bible. It's a lot of fun. I do some debates as well, which you can find on YouTube. I've got a playlist where I keep all the debates I've ever done, whether they be on my own channel or somebody else's. And that's pretty much it. I'd love to do another debate in the future, possibly more specifically about the theology, maybe a Noah's Ark focused debate. That might be a lot of fun. So yeah, we'll see what the future brings.
Awesome. Thank you for that. And then Austin, go ahead and finish it out for us.
So my name's Austin Sims. I run a very rinky-dink channel called Scientific Origins According to Austin Sims. I'm working currently in the field of biostatistics, biometrics, bioinformatics, and technology. I love science, and so I gotta give props to Matt. I am also all about spreading information and knowledge. I don't have any memberships. I don't have any costs. Everything I do is for free, and that's why I put very few resources into it. A lot of times I'm streaming on just my phone and just talking. My goal is to just get as much information out there to help kind of rehabilitate public scientific acumen. And I don't just bust on young Earth creationists. I also talk about anti-vax, flat Earth. I talk about moon landing denial. I talk about pseudo medicine like homeopathics and fake wellness industry stuff that doesn't actually work and doesn't do anything but drain your money, like chiropractic medicine. It's just a general debunking channel. I'm very upfront about the sources I use, and I've always got information to back it up. I'll probably be posting some sources on this video in the comments once we're done. And if I don't, just come to my channel. They'll all be in the posts section. And yeah, that's what my channel is all about. I'm out here fighting what I call the ignorati, or the cult of ignorance. People who are just maybe most of them, I think, are victims of misinformation channels or disinformation. Some of them are knowingly pushing incorrect information. Some of them are unknowingly pushing it. And so I try to be gentle with humans. I'm a humanist, so I believe in kindness. I hate to say that once or twice I have shouted at a creationist or two, but generally I like to think I'm pretty calm and polite, and I care about people. It's all an endeavor of love for me. It's all about humanity, and so I'm here trying to help people as much as I can. Yeah, the homeopathic medicine. I see somebody in the and so we can agree that some of this stuff is bunk and nonsense. Like Donny agrees that the Earth isn't flat, so we have more in common than we might think initially. And so that's what my channel is all about. That's where you can find me. Come by and subscribe. If you like what I do, I'm not saying no if you want to throw donations my way, but I don't ask for them. I do it for free.
Well, I am at the point where I'm about done asking for them from everybody. I do appreciate all the super chats. It was good, stimulating conversation between everybody here, and I feel like if we had 3 more hours and more time in the day, we would have kept going. But
I'd need another coffee for that.
Yeah, same here. Same here. But it's not because I wouldn't want to do it. All right. Well, thank you guys, and we'll catch you on the next one.
Okay. See you later. Thanks for having us.